welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner.
Okay, good morning. On behalf of the CBER Organizing Committee for the REdI meeting for 2026, I'd like to welcome our in-room and virtual attendees to day one. And they fixed the pointer a second ago and it's not working again. So my name is Dr. Graham Price. I'm the Chief of Gene Therapy Branch 5 in the Office of Gene Therapies within CBER's Super Office of Therapeutic Products. So I'd like to go over a few housekeeping issues before we get started on our program today. So the first thing is our on-site Wi-Fi is FDA guest. Now we've got organized an interesting series of sessions for the next day and a half, with speakers from all three of CBER's product offices, as well as from the Office of Biostatistics and Pharmacovigilance, and from the Office of Compliance and Biologics Quality. For our online attendees, please take advantage of the chat pod to put your questions for speakers into the chat pod so that they can be asked in the room. For the in-person attendees, please come up to the two microphones we have in the room at the end of the talks, and we will take questions for our speakers. So the talks will be 30 minutes long, We have 10 minutes for questions. Once this session is complete, then we'll break for lunch and return at 1:30 p.m. So if everything runs to time, we'll have plenty of time for getting questions addressed, and I hope you enjoy our sessions. So I'm going to stand here looking foolish for a couple of moments while we get the pointer and slide advancer reconnected. Again, to our online attendees and to our in-person attendees, welcome to day one.
Let's take a look here and see.
It was working 2 minutes ago.
I believe you. It knows whenever you're on mic too. So what we'll do is I'm going to show them how to do it on their side,
right,
and then I'll get Adam involved and let them.
There's
nothing here on the side usually, so I'll go get that.
Okay, so it looks like we've got our technological difficulties resolved. Again, I'd just like to go through a few more of the details and logistics that apply to everyone. Attendees will be given session evaluations. Please fill these out. They all work today, and this will contribute to our overall evaluation and give you a certificate of attendance. The link will be activated and sent in the post-event thank you email. Please note that the evaluations are only going to be available for 2 weeks, with it closing on June the 3rd. And this is very important for us as it helps to plan future meetings and work out what you all are interested in so that we can arrange the agenda for next year. Recordings of the event will be posted to the web
for drugs and biologics, the 2020 guidance on interacting with the FDA on complex innovative trial design, the ICH E9R1 expert group working group on estimates and sensitivity analysis, and the FDA draft guidance on the use of Bayesian methodology in clinical trials of drug and biological products. Dr. Scott holds a PhD in biostatistics from the University of Pittsburgh, a master's in mathematics from Washington University in St. Louis, and a BA in liberal arts from Sarah Lawrence College. He's a fellow of the American Statistical Association and is a past editor of the journal Pharmaceutical Statistics. Please, Bob.
Thanks, Osman. So it was touch and go with the AV, but I guess we're set up. I need to give people a shorter bio. It's embarrassing. So I'm going to be leading off today talking about FDA's new draft guidance on the use of Bayesian approaches for clinical trials of drugs and biologics. Typically, starting a track with a statistics talk would be seen as a punishment for the audience. I'm going to try to explain why this is important and how it might be useful for some of you. So I've worked on a lot of statistical guidances and other projects at FDA over the years, and this one by far has received the most media attention following publication. I have just a few examples here. This is from Undark, Why the FDA is Embracing Old Math for New Drugs. This one is from Medscape, FDA Changes Drug Trial Guidance. Experts Take Sides. This one, is sort of like a pro and con. At the time I screenshotted it, it had 76 thumbs ups. I don't know if that's a lot of thumbs ups, but let's say it is. And then my personal favorite, the first time I've seen a clickbait headline for an FDA statistical guidance, one weird thing that's been holding drug trials back. The one weird thing, I guess, is frequentist statistics. So what am I going to be talking about today? I'm going to start with probably about half the time talking about Bayesian statistics in general at a very high level and why it matters. And then I'm going to talk about FDA's draft Bayesian guidance for drugs and biologics. And the guidance is a fairly long document. I'm going to be focusing really on just the first few pages where we walk Bayesian statistics can be used for describing data, for drawing inference from a sample to a larger population or universe, for making predictions, and especially for guiding decision-making in a quantitative way. The one thing that all Bayesian approaches have in common is that in some way they're using Bayes' theorem. And what Bayes' theorem is a mathematical tool for combining existing information, things you already know, with new data that is, in this context, would be a new clinical trial, but in other contexts could be anything, to get an updated sense of what you now know once you combined what you knew, what you collected. Now you have an updated sort of state of belief or updated information that you can then base decision-making on. So this is very broad. Not every Bayesian analysis follows these principles, but this is sort of the uniting philosophy to a lot of Bayesian approaches, is three principles. One is evidence synthesis, which is that in general, when you're doing an analysis, you should be incorporating all relevant information in your conclusion. So in particular, that might mean not just the information from the trial or the experiment you just ran, but things you knew before that as well. Also, I do want to stress, it is all relevant information. So one of the things that people will sometimes object to with Bayesian statistics is they'll say, well, you can cherry pick favorable information, bring that favorable information into an analysis and make things look better than they are. It is very important not to cherry pick and to really understand the entire landscape of information before you do this. The second principle is continued or continuous learning. So one of the things that's neat about Bayesian statistics is that kind of at any time, you can update your information by combining the data collected to date with your prior distribution. You get an updated understanding, and then you can collect more data and update again and learn in a continuous way. And then finally, decision-making. A lot of Bayesian approaches have a real emphasis on making optimal decisions.
Then you say if the data are too
unusual under the null assumption, we'll conclude that the null hypothesis is probably false. And if the null hypothesis was the drug doesn't work, and you say it's probably false, then you say we can conclude the drug probably works. So that's the logic of frequentist hypothesis testing. It's important to note that it's working backwards, right? You want to know things about the efficacy of the drug. You start by assuming a lack of efficacy and comparing the data to that assumption. So what's different about Bayesian statistics? So I set this up sort of point by point. So frequentist statistics, again, you're looking at probability distributions associated with the data under the assumption of a hypothesis. So you're saying, what's the probability of the data I collected? And you assumed the hypothesis. The Bayesian approach says, we've got the data, so conditional on that data, what's the probability that the hypothesis we care about is true? So it's forward inference. It's what we actually care about. Frequentist approaches generally start with a blank slate for each experiment, which some people consider an advantage, that you're coming in sort of open-minded with no prior beliefs, and you're saying, what do the data tell us from this experiment? Bayesian approaches, again, will typically start with existing information and update that information. And frequentist approaches typically focus on inferring a population quantity from a sample. So you imagine that there is some average treatment effect for all patients with a given condition and all future patients with a given condition. And what you have is a small sample of those patients. And you estimate in that sample and infer to the population. Whereas Bayesian approaches will often focus instead on decision-making, not I'm going to tell you whether or not the quantity in the population is above such a number, but more, is it an overall good if we make this decision versus that decision? So I do want to qualify what I said a little bit. So the Bayesian approach, the first point, you're getting the probability that a hypothesis is true, conditional on the data you observed. It is what you care about. The cost of that is you have to have a prior distribution. You have to think about everything you know and summarize it accurately. So why do Bayesian approaches matter? None of these things is always true, but in general, using a Bayesian approach can yield more informed decisions because you're incorporating more of what you know in your analysis. In some cases, it can mean that you get to decisions faster. So if you think about statistical power, which in a phase 3 study is going to be related to your sample size, you think about a Bayesian approach where you start with some stuff you already know, you might be able to reduce that sample size by not sort of starting from scratch. In some cases, Bayesian approaches can be used to address more complex scientific questions. So one of the main applications we see for Bayesian approaches is in complex adaptive clinical trials. You can do adaptive clinical trials without being a Bayesian, and we do them all the time. We do things like stopping early for overwhelming evidence of efficacy or for futility, or reevaluating a sample size, or in some cases, starting with 2 doses and picking one halfway through and carrying it forward. Where Bayesian approaches really shine is when you want to do a bunch of those things at the same time. That's very hard to do mathematically as a frequentist. It's kind of not that hard to do as a Bayesian. So all of these things add up to a potential to improve the efficiency of product development. Again, I don't want to oversell it. It's not going to be true in every case, but in some cases, Bayesian approaches can really get you to your answer faster. Okay, so moving on to describing the guidance itself. So the title, Use of Bayesian Methodology in Clinical Trials of Drug and Biological Products. So drug and biological, this is a CBER joint project. Yeah, so it was, I think the Federal Register notice was at like the very end of December, and then this guy has a January 2026 date. Basically, it was a New Year's present. So it was published just a few months ago, but it was really the culmination of many, many years of building up to this point. For one thing, it fulfilled a commitment we made for PDUFA 7, which started in 2022, explicitly to write and publish this guidance. But even before that, we had commitments from the 21st Century Cures Act and PDUFA 6 that had us writing adaptive design guidance, complex innovative design guidance to touch on Bayesian topics. And we've been thinking about Bayesian methodologies for a long time before that. The first CBER Bayesian approval was back in 2008. And in 2010, CBER and CDRH published a Bayesian guidance for medical devices. So we're 16 years later to get it out for drugs and biologics, but it's also a very different guidance. It has very different advice. So the current status, we're considering the public comments we received in the first few months of the year. And we have a commitment in PDUFA 7 to finalize this ideally by late next year. So hopefully that will happen. And I'll say just like a high level overview, we got a lot of really helpful public comments, some positive, some negative, but mostly people were very happy that FDA was providing a framework to think about how to do these things. So the topics covered in the Bayesian guidance, the first topic is areas where Bayesian methods have been used in the past for drug and biologic trials, which are, can be seen as potential application areas. The next topic is operating characteristics and decision criteria. So operating characteristics of a clinical trial sort of refers to how likely it is to get you the answer, the correct answer. In frequentist statistics, we talk about type 1 and type 2 error, which is sort of, I'm not going to describe exactly what that is. In Bayesian statistics, we might talk about a false positive probability or a false negative probability, that is, concluding an ineffective drug is effective or vice versa. And decision criteria refers to the specific mathematical rule we set up to make that decision. The reason this is an important topic for Bayesian statistics is in the frequentist world, you still have to know your operating characteristics, but these questions are sort of already answered by established practice. The decision criterion for a phase 3 frequentist trial is almost always a significance test with a significance level of one-sided 0.025. You might be familiar with p-values, p less than 0.025. When you're doing Bayesian statistics, there is no one rule, and you have to really think carefully about it on a case-by-case basis. I think probably the bulk of the guidance talks about prior distributions from a mathematical perspective, different kinds of prior distributions and how you construct them. And it's a very important topic when you're doing a Bayesian analysis. It's probably the most important topic. And then a few additional smaller but important topics, which is what estimands mean in a Bayesian context, software and computational considerations, and documentation. I didn't include like a history of Bayes in this talk, but one of the reasons, so Bayes has been around since the 18th century. We had that lovely picture of the Rev. Thomas. One of the reasons it's only now that we're talking about moving it in a serious way into clinical trials for drugs and biologics was throughout most of the 20th century, the computational burdens of doing Bayes were prohibitive. You have to do a lot of computer work to get to your answer. And even as, well, I'd like to say it was recent, even as recently as when I was a grad student, it could be, days or even weeks to do certain kinds of computations. That stuff is basically trivial now, at least for clinical trial type questions. There's nothing you can't do in a minute or two at the worst. And then finally, documentation. How do you document a Bayesian proposal, a Bayesian protocol, and a clinical study report that includes Bayesian analyses? All that said, for this talk, I'm really going to be focusing on this first one, the potential application areas. So the first application, and so this is the definitely like the catchiest, the most exciting. It's not necessarily the most applicable to every program. This is something that FDA discusses with sponsors on a real case-by-case basis, but when it is appropriate, it can really change the landscape, which is borrowing information from previous trials. So the idea here is that maybe you're running a phase 3 study, maybe it's not your first one, or you have a randomized phase 2 study, and you want to use the information from the previous clinical trial in the design and analysis of a new phase 3 trial. So people use the word borrowing for incorporating the information from the previous trial. It's not a great word. You don't give the data back. You just use the data. But you'll see the word borrowing. You'll also sometimes see the word leveraging. So in some circumstances, this strategy can be used to improve both the quality of the information we get from a trial, because rather than starting from a blank slate and pretending we don't know anything, we're getting an answer that incorporates all of the controlled information we have to date, and also the efficiency. So this really can reduce sample sizes in some cases. It requires very careful planning and typically a lot of discussion with FDA. The next example is probably a little bit more widely applicable and very related, which is augmenting control arms with external data. So this is I like to think of this in the context of rare diseases. So if you're doing a confirmatory trial for a rare disease, a randomized controlled trial, often you won't know much about the treatment effect going into that trial. But if the control is standard of care, you might know quite a bit about likely outcomes for control patients. You might have registries, natural history studies, earlier studies of the same agent, and you can sometimes leverage that control information to beef up the control arm in your phase 3 study. So the way you do this is you construct a prior distribution that incorporates relevant external information about the control. And again, that could come from a previous trial or registry natural history study. So the nice thing about this is it can reduce the required number of participants randomized to control, which overall reduces the study size and maybe time. Also, in some settings, there's squeamishness about randomizing a lot of people to control if you think your investigational therapy is likely to be much more effective than control. Pediatric extrapolation is a very important topic, and not all pediatric extrapolation is Bayesian, but the Bayesian framework is a really kind of straightforward way to do this. So the basic idea here, when FDA approves a drug or a biologic for adults, there's PREA that kicks in, and in most cases, the manufacturers have to initiate studies in pediatric patients as well to determine that it also works for children and is safe for children. So what you can do with Bayesian statistics is borrow some of the information from the adult trials that were used to show efficacy in adults to help kind of reduce the size and the number of pediatric patients needed for the pediatric trials. When people talk about PREA, one of the things they say, one of the reasons we have it, is that children are not small adults, right? If something works for adults, you can't just always assume that it's going to be exactly the same for children. But in this context, I like to say children are human beings. So if you have a product that is highly efficacious in adults, it is unlikely that that information is totally irrelevant from children. And so you can use extrapolation in some cases to at least give you a head start toward the efficacy study in children. Another example is borrowing information across similar diseases. So there are a lot of cases where this can come up. We've seen applications in chronic pain, which can occur for a variety of reasons. Another example would be different cancers of different systems, but where the tumor shares a molecular signature, especially if that signature is what's targeted by the intervention. And in these cases, in some cases, you can use Bayesian methods to share information across those multiple diseases. One case where this can be used is in basket trials where you're already looking at the multiple potential indications in the same trial, and it's natural to kind of leverage to some degree the information. Borrowing information between subgroups. So when we do a phase 3 clinical trial, we get an estimate of a treatment effect in the entire trial population, which we hope is very close to the planned indicated population. But it's also always important to understand what's happening in important subpopulations of patients, getting more toward personalized medicine. How does this work for somebody like me? So you might look at demographic characteristics. You might look in males versus females, older adults versus younger adults. You might look at different clinical characteristics, severity, things like that. And one way to do that is just to say, well, in my study, this was the average effect in males, and this was the average effect in older adults, and this was the average effect in patients with mild disease. And that's a natural way to estimate it, but statistically, those estimates can be very unstable because the sample sizes are small. You're doing so many of the estimates that in general, it's known that if you tried to replicate the study, the next answer you got to the same question might be quite different. So there's an approach called shrinkage estimation, which also is not always Bayesian, but Bayesian's a natural way to do it, where you say, okay, my subgroup effect, I have an effect for males and I have an effect for females. And then I have an overall effect for everybody. And my estimate for males is going to be somewhere between the overall effect and the study specific male effect. So you're shrinking everything a little bit toward the average. And that kind of estimate tends to have much better statistical characteristics. And then the last thing I'm going to mention, dose finding in oncology. So this would not generally be in phase 3. This would be in phase 1 dose escalation trials. I mention it because it's one of the most widely used applications of Bayesian methods. And people have been doing this for 10, 15 years in CBER and CEDAR. The basic idea, people who work in oncology, well, you may already be familiar with this, but you also may be familiar with things like the 3+3 design. The basic idea is to kind of titrate dose to toxicity, but Bayesian methods let you do it in a very precise way where as patients experience toxicity, you take the dose down. As enough patients don't experience toxicity, you bring it up a little bit, and you can get to a very precise estimate of the maximum tolerated dose, which you would then take into phase 2 in most cases. Okay, I have 3 minutes left. My timing's better than I thought. I'm going to talk about one example of Bayesian borrowing across trials, which is a product called REbiota. So REbiota is a fecal microbiota transplant that was approved by CBER in 2022, and it was indicated for the prevention of recurrence of Clostridium difficile infection following antibiotic treatment for recurrent CDI. So people may already be familiar with CDI. CDI, terrible diarrheal illness is associated with it. It's often something that occurs after treatment with antibiotics for another infectious disease. Your sort of healthy gut flora get wiped out, the Clostridium difficile come in, and you can get into a really bad cycle where then you have antibiotics to get rid of the C. diff, which leads to being colonized with C. diff again. And in some patients, it can be debilitating and fatal in a lot of older adults in particular. And so fecal microbiota transplant, the idea behind this modality is to transplant healthy gut flora from donors into patients who are having this experience. So this product was approved with primary evidence of effectiveness coming from a Bayesian analysis that borrowed phase 2 data into phase 3. So they had a positive phase 2 randomized trial, but it didn't sort of pass the p less than 0.025 threshold. They were doing a phase 3 study, but it was not going to be possible to complete the study, in part because fecal microbiota transplant was available off-label at a lot of academic medical centers. So instead, they did a Bayesian analysis where they borrowed the phase 2 data into the phase 3 analysis. I'm not going to talk about this in great detail, but this is an example of a posterior distribution of a treatment effect. So what the posterior distribution tells us in this case, the sort of shaded in part of the bell curve, forget the colors, all the shaded in parts of the bell curve, you look at how much of that mass is to the right of 0. And that's the probability that the treatment effect is greater than 0. And if you add it all up, it was 99.1% probable that the treatment was more effective than nothing. And one of the nice things about Bayes is you can look at this distribution and answer a lot of other potentially clinically relevant questions. Say, for example, what's the probability that the treatment effect is greater than 10 percentage points, which in this case was 71.5%. So that's it. I have some links here, a link to the guidance, of course, and some related material. So in summary, Bayesian statistical approaches can offer the potential to increase efficiency and in some cases the usefulness or effectiveness of clinical trials. There are a lot of potential applications that range from borrowing information from external sources to extrapolating from adults to children to getting more accurate estimates of subgroup-specific treatment effects. And our draft guidance, I guess, I'm still not sure if it should say 2025 or 2026. I'll fix that the next time I give this talk. Our draft guidance on the use of Bayesian methodology in clinical trials of drug and biological products describes FDA's current thinking on this topic and provides a framework for how to develop proposals and interact with FDA on Bayesian proposals. So we'll stop for questions with 7 seconds left. Amazing.
First of all, thank you so much for this talk. I'm geeking out right now because my entire focus is in adaptive platform trials and work very closely with Don Berry and Scott Berry, who you probably know. I would say over the last few years, and I'm hoping this will improve based on what we're seeing today, it does seem like the agency had historically had a bit of a challenge accepting Bayesian statistics in adaptive platform trial settings, particularly in more rare diseases like where I focus is myeloma. It's really important to be able to, there are so many new therapies, particularly in biologics. And so we use these as a mechanism to study these therapies. Do you anticipate first that the agency will bring more Bayesian statisticians into the submission review process, and even as early as PIND and IND review? And then second question is, what is your position on historical or synthetic controls, especially in this particular marketplace? It's so important to be able to look at these things quickly.
Mm-hmm. Sure, yeah. The first question is easier than the second. The first question is yes. There will be more Bayesian statisticians engaged in review. There's kind of just a basic secular trend reason for that, which is that there are more Bayesian statisticians. It's when I started, my training was both frequentist and Bayesian, and I was kind of the Bayesian in CBER when I started. And now increasingly, it is much more common for people to come out of biostat grad programs with really sound Bayesian training. And so there's more people, but also because of the guidance and because of training we're offering, I think a lot of people are getting more up to speed on how these methods can be applied. And as I'm sure you know, there have been some extremely clever and important adaptive platform trials that have used Bayesian methods. The second question is tough. I want to say case by case. I think in particular for adaptive platform trials, I have to say it's more of a CDER question than a CBER question. We have relatively few platform trial proposals at CBER. I think there's been just a constant increase in the interest of using historical controls in some way in the trials. I think people are often more comfortable using them to augment than to replace a concurrent control. Synthetic controls, personally, for me, it's usually is the juice worth the squeeze situation, because I think I don't want to be too negative because it might be overinterpreted, but it's not always clear to me that if you have enough information to do a good synthetic control analysis, then maybe there was another way to leverage that information. But that's just my personal opinion. Yeah.
Yeah, there's an online question. Can a Bayesian approach be utilized in biosimilar development? and how can it be applied in biosimilar clinical trial design? Because, for example, one presentation has been established through phase 3 study. How can Bayesian methodology be used to inform development of a higher dose presentation?
Also a good question, and also a question better for a CDER speaker, because the biosimilars are by and large CDER products. I can only answer it in a general way, which is, I think, yes, Bayesian methods are potentially applicable the same way they're applicable in other areas, and would definitely encourage you to talk to the review team about it if you're working on a biosimilar new dose proposal.
My question is about the data submission package and how use of the Bayesian methods affects the data submission package.
That's a good question. So the reason that question might come up would be that your prior distribution incorporates previous information and, for example, is raw line data expected for that previous information. I can't honestly say we have a clear answer to that so far. In the example I gave you from REBIOTA, it wasn't problematic to include the raw line data because it was a clinical trial from the same sponsor. In cases where the information is coming from natural history study or registry study, I think probably we would try to work with the applicant to have as much data as we needed to verify that the prior was constructed properly. But the prior itself is not data. The prior is something that's been informed by existing information. So again, case by case, but it's a great question and something we're considering in the revision. Thanks.
If Bayesian approaches are useful, why are they not used all the time?
Great question. So, yeah, and it's a question I'm always asking everybody. No, there is a pretty straightforward answer to that, which is to do Bayesian approaches properly, you need to have a really good and correct prior distribution, and that is work. It is work. People have to discuss it a lot. My prior distribution might not be the same as an applicant's prior distribution. And for the regulatory decision, well, I'm not going to say it's my prior distribution that matters, but it is FDA's prior distribution that's going to matter. And so if you can't come to agreement on that, then it's difficult to move forward. So there's a lot of overhead in using Bayesian approaches. It's also can be computationally difficult. But I think over time, we're going to see more and more use of Bayesian methods. I think some of it is just inertia, doing the thing that's been done. And once people started, broadening that out to include more Bayesian approaches, we'll see more people following suit.
Questions from the audience? My question?
Can Bayesian methods be used in drug safety assessment? Would you know some examples?
Yes. Can Bayesian methods be used in drug safety assessment? Absolutely. There's no reason why not, but drug safety assessment presents a whole host of statistical issues that don't go away because you're being a Bayesian. In the simplest case, if you're looking at a safety trial where there's a defined safety endpoint, then everything we say in the guidance about efficacy would also apply to that safety trial. But in the more common case where safety means looking at potentially unexpected adverse experiences that occurred during a trial, there's always a risk of sort of over-interpreting things that might not generalize to a population. But I will tell you an example where we used a Bayesian safety analysis. Actually, two examples. The 2008 CBER approval that I mentioned that used a Bayesian analysis was for a factor VIII product for hemophilia A, and the primary endpoint of that trial was a safety endpoint. It was inhibitor development. Another example, the hepatitis B vaccine, Heplisav, during the development of that product, there was an unexpected increase in the occurrence of MIs in the experimental arm versus control, which was a different hepatitis B vaccine. We took this issue to an advisory committee, and part of that discussion included a Bayesian analysis to try to say where do we think the probability that this MI risk is real might lie based on the data we've seen. So in that case, it was being used more as an exploratory analysis.
One more question. At what point in developing a Bayesian approach should an applicant contact CBER?
Yeah, so even in the plenary session, people were saying, we don't want to say talk to us early because it's a cliche, but talk to us early. Whenever you're considering using a Bayesian approach in a confirmatory or pivotal trial, interaction with FDA is vital. And I think it was Michael Davis who said, it might have been Owen Ferris, one of the two of them said, tell us your story. And that applies here too. Tell us why you're proposing this approach, why it's going to be useful for the program. And that really helps us a lot in understanding how to review it.
One more question.
Hi, I'm Kim from CGT Consulting. I work with a lot of small biotechs, and so I'm just kind of curious. Imagine you're a small biotech, clinical stage. How would you get started with this type of approach? What type of computational tools or applications would you use to generate this prior distribution, et cetera?
It's a great question. I mean, so a lot of people will turn to expert consultants in Bayesian statistics or CROs that have Bayesian expertise if they don't have it in-house. Obviously, best case is you have a Bayesian statistician in-house. In terms of software, there are a lot of different packages. I always bump up against the sort of principle that FDA shouldn't be endorsing specific software, but there are free tools for doing Bayesian analysis, as well as a lot of off-the-shelf commercial products. So the math of it for a working statistician is not really difficult. It's more the conceptual process of planning the Bayesian analysis, figuring out what information is relevant and how to use it, and sort of working with FDA on those questions, I think. Yeah. So, sure, of course.
Our second speaker for our session is Dr. Honyan, who's my long-term mentor and an acting director of Division of Analytics and Benefit Risk Assessment in the Office of Biostatistics and Pharmacovigilance. She's an expert in quantitative benefit risk assessments and have led benefit risk assessments in CBER to inform regulatory decisions. She is active in research and collaboration to develop novel benefit risk approaches, as well as training and outreach for implementation of the benefit risk framework. She was a member of FDA Benefit Risk Guidance Working Group, as well as FDA's Benefit Risk Framework implementation team. She made major contribution to a report by SIOMS, Council for International Organizations of Medical Sciences, for benefit risk balance for medicinal products. She is actively engaging with multiple working groups of professional societies, including American Statistical Association and International Society for Pharmacoepidemiology, in activities of benefit risk assessment planning, methodology, and tool development. Please welcome Dr.
Thank you, Osma, for the introduction. So a lot of time when we go to conference and talk to someone from outside FDA, one question come a lot is say, we know a new drug could save life, but there is also carry some risk. So the question is how FDA decide if it was to approve a new drug. So in this section, basically, I will mainly talking about how FDA conduct benefit risk assessment to inform regulatory decision. So outline of my talk will have 4 topics. First, I will discuss the regulatory context for benefit risk assessment. And then I will discuss FDA's benefit risk guidance. And then I will turn to discuss more specific benefit risk assessment consideration for biologics that are regulated by the CBER. And then last, I will talk a little bit about how the additional quantitative analysis can be used to inform benefit risk assessment and maybe provide an example. The Food and Drug Cosmetic Act was enacted in 1938. And the statute require FDA for approval of the licensure, require FDA to establish substantial evidence that the drug will have efficacy and will be efficacious and safe for its intended use. In this statute, there's no clear guidance about what is considered as safe. FDA's benefit risk guidance provide a further interpretation. And the guidance state that for a sponsor try to apply for the product licensure, you need to provide a substantial evidence to indicate the benefit of the products outweigh its risk for intended use. Now, I would like to give you an overview of FDA benefit risk guidance. It was published in October 2023 as a part of PDUFA commitment. And the FDA's benefit risk guidance covers 6 major topics. The first topic is discuss about FDA's benefit risk framework and benefit risk consideration for review of new drug application and the biologic licensure application. The guidance also discuss how the uncertainty of benefit risk evidence will impact FDA's decision about the approval. And the guidance discussed when the patient input become important and how the patient preference data should be collected to support benefit risk assessment. FDA's guidance taking the FDA's benefit risk framework, taking the life cycle approach, and talking about you need to do the benefit risk assessment throughout product life cycle, and also has a good planning. The guidance emphasizes the importance of early and timely interaction with FDA regarding benefit risk assessment and benefit risk planning. And the guidance also talking about FDA's continued benefit risk assessment in the post-market setting. And this table is benefit risk summary table in the FDA's review template for new drug application and biologic licensure application. In the first column of the table, it summarizes the 4 dimensions of the FDA's benefit risk framework. The first 2 dimensions is analysis of condition and current treatment option. And these two dimensions are disease specific. And then the last two dimensions is benefit and risk management. And these two dimensions is product specific. On the second column of the table is the evidence of, you summarize evidence of each dimension and also record uncertainty. And the last column of the table here and summarize the conclusion of the assessment for each of the dimensions and also the rationale. At the bottom of this row is the overall conclusion for the benefit risk assessment. You integrate all the information from the 4 dimensions and then make final conclusion about the benefit risk of the products. Now I'm going to go over the dimension, benefit risk dimension one by one. And then the first dimension is analysis of condition. So in this dimension, FDA will consider indication of the drug. Basically, what disease and condition to treat, and what are the target population. And then we need to consider disease burden. For example, the incidence of disease among the population, and duration of the disease, and the severity of the disease, and how it impacts patients' life. We sometimes also consider societal and public health implications of the disease. And the second dimension is current treatment option. So in this dimension, we need to consider what are the available treatment options for the patient. And we also need to consider what are the standards of care. In this dimension, we need to consider what are the efficacy, safety, and the burden of the treatment to the patient. What are the limitations for those available options? So main purpose is to try to identify unmet medical need. And also, we will look at if there is any specific sub-patient population, they have specific unmet medical need. And the third dimension is the benefit. So there is a lot of consideration in terms of you consider benefit of a drug. So that is case by case. But here, I just give you a few examples. One thing is very important is to consider nature of the benefit. So for example, if you have 2 drugs, one provide an option of curable option, and then another drug is just to manage the disease condition. Then obviously, when you consider the benefit, and then you will give much weight on this benefit, provide curable option. And we also need to consider effect size of the treatment, also durability of the effect. We need to consider if there is any specific patient population, they are most likely get more benefit with low risk. Are we able to identify those patient population as a target treatment population? We need to consider if the clinical trial data we can obtain from the clinical trial can be extrapolated or generalized to the real world setting. And we need to consider strength of the evidence. And the last the dimension is risk and risk management. And for this dimension, we need to consider severity, likelihood, and reversibility of the adverse effect. We also need to consider known risk. So those risks you observe, adverse event you observe in the clinical trial, but you also need to consider if there's any potential unknown risk. It may emerge after the products enter the market. So we need to consider strength of the limitation of the evidence. Also, we need to consider if there is risk identified, are we able to find any potential risk mitigation to reduce this risk, but at the same time still maintain the benefit of the drug. And then finally, you make conclusion based on all the dimensions we assess. And then we consider what are the key benefit evidence, what are the key safety evidence, what are the uncertainty associated with those evidence, how those uncertainty will impact our conclusion. And we also need to consider, as we discussed earlier, therapeutic context. That is basically what are the severity of the disease and what are the unmet medical needs. And we need to consider how those dimensions will impact our benefit risk conclusion. FDA will consider the benefit risk in overall patient population, but we also consider some individual patients' perspective, especially for some treatment. The patient preference are very different. And then we also, in this part, we need to consider what are the potential risk mitigation we can use to reduce the risk and to support favorable benefit risk. FDA's guidance discussed the impact of uncertainty on FDA's benefit risk assessment. The guidance state benefit risk assessment depend on the strength and quality of the evidence. Many sources of the uncertainty can be anticipated. If with a good study design, we can avoid many of them. And therapeutic context play an important role in the acceptance of the uncertainty. Here I give an example like a vaccine, which is used among the larger health population, is used to prevent disease. Therefore, the vaccine, the tolerance for risk and uncertainty are very low. In opposite, there's some serious disease or some rare disease. They even don't have any other treatment option. So in that cases, many of the patients are willing to take the certain risk or uncertainty in exchange to some clinical meaningful benefit. So when FDA doing benefit risk assessment, some safety issue may be balanced if the data suggests there is a greater uncertainty for some clinical meaningful benefit. The FDA guidance also discuss importance of patient input. Patient input can inform FDA benefit risk assessment. However, the patient experience data need to be collected through scientific sound methodology. Patient input can inform therapeutic context. For example, tell us how the disease impact their life. What are the key benefits they are hopeful? What is their tolerance of the risk and uncertainty? What kind of the trade-off they are willing to do to exchange the benefit with the risk? And what is the burden of the risk mitigation? So FDA will collect patient input information. And then in many of the situations when the patient preference are sensitive to the decision. FDA will balance the perspective of individual patient with the judgment regarding overall benefit risk to the patient population. One thing I need to point out is patient preference information is not meant to substitute for any adequate evidence for effectiveness and safety. And benefit risk planning is very important. Benefit risk planning is a purposeful activity to incorporate benefit risk assessment throughout the drug development life cycle. In the planning, we will identify potential benefit and risk. We will identify what are the primary and surrogate endpoints that can be better used to measure the key benefit and risk in the clinical trial. What are the major target patient population? And what is the risk mitigation can be used to reduce the risk? Design clinical trial with benefit risk in mind will help us to collect good quality data, eventually support benefit risk assessment for products marketing approval. For example, what are the sufficient sample size? What are the sufficient study duration will allow us to observe clinical meaningful benefit? Benefit risk planning is especially important when there is challenge benefit risk issue are anticipated. In that cases, a good study design and good planning will help us to overcome the challenge and to collect data relevant and reliable. And the benefit risk planning will help direct drug development toward favorable benefit risk profile. FDA suggests to have early and timely discussion with the FDA about the benefit risk assessment and planning. FDA's perspective can inform a sponsor's benefit risk planning. Benefit risk assessment planning communication can be occurred in the normal standard meeting with FDA. End of phase 2 could be a critical time point to discuss clinical trial, how to design a trial to collect the data that are strong and have with good quality to inform benefit risk assessment. A discussion before regulatory submission may also helpful because it will allow sponsor understand what kind of the information is needed for benefit risk assessment. FDA's guidance also discuss the discussion with FDA on benefit risk may continue post-market, especially for those products was approved with post-market commitment and requirement. And one thing is very important to understand is FDA's advice about the benefit risk assessment and planning is based on available information provided to the FDA. And the advice may evolve with the new information. FDA's final benefit risk assessment is based on complete information submitted as a part of NDA and BLA. And benefit risk assessment in the post-market setting. FDA take life cycle approach. FDA will continue consider new emerging safety information and evolving therapeutic context and treatment option in the post-market setting. And the post-market periodical reporting is an important mechanism to inform FDA continue benefit risk assessment for approved drug. A formal benefit risk assessment may be conducted by FDA if there is a substantial new evidence indicate there is a potential change of product's benefit risk. Now I would like to focus on a little bit more on the CBER. There is very specific consideration for CBER's products, the biologic regulated by the CBER. The CBER-Regulated vaccine products, blood and blood products, and advanced therapy include cellular and gene therapy. The benefit risk considerations are different, depend on the product. Here I give two examples. One is vaccine, and one is cell and gene therapy. They are at the two ends of a wide spectrum. For the vaccine, it's used in larger health population and is used to prevent the disease. So when we conduct a benefit risk assessment, FDA has a very low tolerance at the risk for the risk and the uncertainty. And we will consider vaccine benefit. We will consider the benefit to individual to prevent the disease. We will also consider the public health benefit that reduce disease transmission among the population. Also, one thing is unique is vaccine efficacy is not directly translated to vaccine benefit. Therefore, you need to consider where the vaccine is used, when, among who. Because the vaccine benefit is not just depend on vaccine efficacy, also depend on disease incidence among the population. So same vaccine with the same efficacy, when you apply to at a different time of the pandemic, at a different location and different population, the benefit will be different. And then in opposite for cell and gene therapy, is used to treat serious or life-threatening disease. Sometimes it's for rare disease, there's no treatment options. And oftentimes, this cell and gene therapy provide the only curable option for the patient. Even though for this cell and gene therapy, they could pose some long-term and irreversible risk. However, because of the severity of the disease, and also a medical need, many patients may be willing to make a trade-off, take a higher risk and uncertainty to exchange for the benefit to treat life-threatening disease. And finally, I would like to talk a little bit about the additional quantitative analysis. So up to now in my presentation, I spent all the time talking about FDA's benefit risk framework. That is a structured framework. So basically, in general, in FDA, the benefit risk assessment follow this structured framework. Consider all the available information, make a judgment. Mainly is the clinical judgment. However, in some situations, when you follow this structured framework, you still have some very critical questions cannot be answered. You cannot make decisions. So in that situation, some additional quantitative analysis may be conducted to inform the benefit risk assessment. An example of the quantitative analysis can do is, one is it can help to integrate multiple benefit and risk. We know for a drug, sometimes they provide multiple benefit and can also pose multiple risk. And then how do you put them together and come up with one benefit risk conclusion? So some quantitative analysis can help to integrate the multiple benefit and risk. Also, a lot of time is very unavoidable to have uncertainty with your benefit risk evidence, your clinical trial data. And then what is the impact of those uncertainty in your benefit risk? And quantitative analysis can help you, and can also help you to understand how much weight, because every patient may have a different preference. Some people like to take more risk, some people maybe not. So people will put in different weight on different outcome. So how those weight will impact your benefit risk decision. And also, you can use the quantitative analysis to evaluate the benefit risk for different subpopulation, because not all the patient population have the same benefit risk profile. And also, we can use quantitative analysis to understand how the data from clinical trial can be extrapolated to the benefit risk in the real world setting. And we can also use quantitative analysis to evaluate effectiveness of the risk mitigation. And here I give you an example of FDA's quantitative analysis for COVID-19 vaccine. And this analysis was conducted in 2021 and published in Vaccine in 2022. As a background, at that time, the excess myocarditis and pericarditis cases associated with COVID-19 vaccine among young men was reported after emergency use authorization of the COVID-19 vaccine. And the challenge posed to FDA is, if this vaccine is still benefit outweigh the risk for this young man with this concerning safety issue. So FDA conduct a benefit risk assessment to evaluate the benefit risk among this age group. This is the diagram we use for the benefit risk assessment. For the vaccine benefit, we consider the vaccine preventable COVID-19 cases, associated hospitalization, ICU stay, and death. And then for the risk, we consider excess myocarditis cases and the hospitalization and the death. At that time, there is great uncertainty for a lot of information. For example, vaccine effectiveness in real world, there's not much data about the vaccine effectiveness. And there's uncertainty about the trajectory of the pandemic. Also because the small number of the cases report for myocarditis cases and death rate is very uncertain. So FDA developed 7 different model scenarios, try to incorporate those different uncertainty. And here I present 2 primary scenarios. One is perspective scenario, use COVID-19 incidence data report in July 10, 2021. And the vaccine attribute myocarditis case rate, death rate is 0.002%. This is the upper bound of the death rate based on the data we got. And then most likely scenario, we assume the incidence were going up, and then we assume the death rate is 0. That actually is because at that time, there's no death was confirmed associated with vaccine. So from here, you can see the result on the, On the left-hand side, this blue bar is represent the benefit. And then the right-hand side, the orange bar is represent the risk. So this is for another pessimistic scenario. So FDA conduct this analysis and report the result, represent the benefit and risk side by side. So this facilitates the discussion for the benefit risk of COVID-19 vaccine. So the key takeaway for this presentation is FDA used benefit risk framework to guide the product review. And sponsors' activity throughout the drug development will help build evidence to support the final benefit risk decision. Purposeful planning of appropriate study design ensure generation of relevant and reliable evidence. Early and effective communication with FDA regarding benefit risk may be helpful. And in some complex cases, additional quantitative analysis may answer critical questions to support benefit risk assessment. I conclude my presentation, and for any question, I would like to answer.
This was a wonderful talk about benefit risk, and you stress quantitative a lot, quantitative. I want to push back and say the benefit risk assessment is not very quantitative. There are some elements that goes into it that are quantitative, but the overall assessment of making a determination of the benefit risk, from my perspective, is qualitative.
Right.
Can you comment on that?
Yeah, I can. Actually, on my presentation, the major part I'm talking about FDA structured benefit risk framework. That is not quantitative. So that is framework, you consider all the dimensions. So that is basically, you take all the information and then you integrate them, thinking through it, and make a clinical judgment. So that is not quantitative. So I think I actually only spend very small part talking about quantitative, additional quantitative benefit risk assessment. And that quantitative, maybe I can put out the slide because I skipped this slide. So I think this slide maybe answer your question. So when I'm talking about quantitative analysis, this is not required for every benefit risk assessment. This quantitative benefit risk assessment is only used to support overall benefit risk assessment framework. And then it is only needed when there is some remaining question cannot resolve. And then quantitative approach can be used to help answer your question. So in that cases, you can have this additional quantitative benefit risk assessment to support your benefit risk framework. And so in most of the cases in FDA, you are definitely correct. We are not doing quantitative benefit risk assessment. Only very few cases, there is some very specific challenge issue cannot be resolved just based on the framework.
What are the common statistical methods and approaches which are used in benefit risk analysis?
So I think it really depends on what are the critical questions. So, so let me go back to the slide early. Yeah, here. So it really depends on what is your So that is the risk part of the donor deferral policy. So for the blood products, we have this kind of the consideration. We also conduct some analysis for that.
Thank you so much, Dr. Yan, for the presentation. Thanks.
Hello, good afternoon. Welcome to session 3. My name is Shoujian Li, and I'm the Deputy Division Director of Biostatistics in CBER's Office of Biostat and Pharmacovigilance. And I will be the moderator for this session. So as you know, the digital health technology and the data collection is crucial for the success of many modern clinical trials. So because it's not only the convenience, but also includes several benefits. For example, broader participation, people who could not join before due to travel distance or busy schedules, now they can. And also better retentions, because the participation is easier, so fewer people drop out. And the more accurate data, frequent continuous measurements improve the precision. And because of these benefits, and we can think about that clinical trials can get a more realistic picture of how a treatment works in patients' everyday life, whether at home or the daily routines, and something that clinic visit alone cannot provide. So I have the pleasure to introduce you our session speaker, Dr. Ujwani Nukala, and she will be talking about this topic, and she will use the electronic diaries as a case studies. So Dr. Nukala serves as a reviewer and a researcher in the Division of Analytics and Benefit Risk Assessment in CBER's Office of Biostat and Pharmacovigilance. She is responsible for reviewing digital health technology, quantitative benefit risk assessment, and the real-world evidence. Dr. Nukala joined FDA in 2019 as an ORISE fellow working on QSP mechanistic models of immunotherapy, and later in 2022 joined Office of Biostat and Pharmacovigilance as a reviewer. She has the master's degree in biochemistry from Osmania University in India, and a second master's degree and a PhD degree in bioinformatics, both from University of Arkansas. Without further ado, let's welcome Dr. Nukala.
Thank you, Shoujian, for the introduction. Good afternoon, everyone. As the famous saying goes, the world is run by those who show up. So thank you all for joining today's session, either in person or online. I'm Ujwani Nukala. I'm a reviewer from CBER Center for Biologics. In the next 30 minutes, you will learn how FDA thinks about using digital health technologies in clinical investigation with a special focus on electronic diaries. Before I begin, please note that the views presented in this presentation are entirely mine. Any regulatory actions must be based on official FDA policy and communications. So here, let me walk you through what we aim to accomplish in this presentation. So by the end of this talk, you should be able to define what a digital health technology is and know their role in clinical investigations. Understand FDA requirements for DHT use in clinical investigations and what are the key considerations for implementing e-diaries in vaccine clinical trials, and also recognize the best practices for ensuring data quality and integrity. So what are digital health technologies? As defined by the FDA, digital health technology is a system that utilizes computing platforms, connectivity, software, and/or sensors for healthcare-related purposes. The definition is broad intentionally, and it covers from a general wellness application on your mobile to a sophisticated medical device such as continuous glucose monitor. So in the clinical trials, we are using these DHTs to collect data from the patients or the participants remotely in their everyday environment rather than from a clinic visit. So this gives us a much richer and clear reflection of how a participant is feeling in their day-to-day life rather than from a single clinic visit. So if you think about it, instead of asking a person how they felt yesterday or a week ago, we are collecting the data as it happens, whether they are at home or at work. Some of the examples of these digital health technologies include sensors in wearables that can track your steps, software on a smartphone to conduct cognitive tests, and continuous glucose monitors that monitors your blood glucose levels continuously. So this shift from episodic to continuous data collection is a significant change in how we are running the clinical trials today with the help of these digital health technologies. So in December 2023, FDA has published this final guidance document, Digital Health Technologies for Remote Data Acquisition in Clinical Investigations. And this document will be the reference for what all we are discussing in this talk. And this document was co-developed by CBER, CDRH, and Oncology Center of Excellence. And this shows the cross-cutting nature of DHT use in the development of different medical products. And this guidance covers the entire life cycle of a DHT, from selecting an appropriate DHT to validating and verifying it and maintaining it throughout the life cycle of the clinical study. And on the right, you can see the screenshot of the FDA's DHT webpage. This could be your go-to resource to know more about the DHT programs and the contact information if you have any questions. So how does the FDA regulate DHTs? So in clinical trials, data integrity is governed by ALCOA plus principles. And here each letter represents a specific requirement. So when DHTs are used to collect data in the clinical trials, the data should be attributable, meaning it should be clear who documented the data, and it should be legible, meaning it should be readable and understandable, and it should be contemporaneous. The data must be recorded in real time or near real time, and it should be original with records of who updated and when, and it should be accurate without any errors. Coming to the plus principles, they show that the data should be complete, consistent, enduring, and available for the review and auditing. So the key message here is DHTs, when they are properly implemented, they can meet all these ALCOA plus principles. And another goal is to protect patient safety. So when these DHTs are not used properly, they can pose some risks to the participants. So the FDA's approach is built around a concept called fit for purpose. This is the conclusion that the level of validation associated with a DHT is sufficient to support its proposed use. FDA focuses on ensuring that the DHT is appropriate and validated for its specific purpose. So we will learn more about how we can make sure that the DHT is fit for purpose in the next few slides. It is important to note that FDA does not regulate the technology itself, or neither it certifies or dictates which DHT to use in the clinical trials. But FDA evaluates if the chosen algorithm or the technology is reliable, is it appropriate, and is the data collected complete and accurate, and does it support regulatory decision making. So in order to verify if the DHT is fit for purpose, verification and validation are two important processes. These two are distinct processes but are complementary. In verification, we ask if the DHT measures the clinical parameter accurately and precisely. For example, is the given thermometer actually measuring the temperature within the ranges that is claimed? Whereas in validation, we ask, is the DHT appropriately assessing the clinical event in the proposed population? For example, a step counting algorithm which was validated in a healthy population might work accurately, but it may not work accurately in an elderly participants with Parkinson's disease. So this will be a validation problem. And these verification and validation processes may begin with benchtop studies to test the technical performance of the device, and it progresses to healthy volunteers to test in human use conditions. And ultimately, they have to be tested in the target population to make sure the DHT works as intended where they matter the most. And usability evaluations. So these usability evaluations answers a simple question. Can the participant use the DHTs in a real world without any errors that could compromise the data quality and or their safety? So these usability evaluation must be tailored based on the risk and the complexity of the device. Some of the elements these evaluations must look into are the ease of navigation and is the login process easy enough for the participants, and are they able to complete the tasks efficiently, and is the participant able to understand the instructions, and do they know how to respond to alerts and notifications, and how are they feeling about the technology? Are they comfortable enough? So all these evaluations must be undertaken, and the findings must be fed directly into the design improvements and into the instructions for use and in participants' training materials. So the purpose of these usability evaluations is to iteratively improve participants' experience until they can use these DHTs independently and correctly. So the key message here is even the most accurate, well-validated DHT is useless if the participant cannot figure out how to use it correctly. So like with any other technology, DHTs may pose some risks to the participants, which may be broadly categorized into clinical risks and privacy-related risks. Coming to the clinical risks, the participants might have physical discomfort or injury. However, by evaluating the design of the wearable and providing sizing options, some of these risks can be mitigated. And with rigorous validation and calibration checks, erroneous measurements can be avoided. And with regular alerts and clear protocols, delayed response to any adverse reactions can also be mitigated. And robust security measures must be in place to avoid any cybersecurity threats. And in clinical trials, we are using these DHTs to collect very sensitive health information from the participants. So it is very important to protect this data from any data breaches, unauthorized access, and any third-party data sharing. So these risks can be avoided with proper encryptions or multi-factor authentications and comprehensive security measures. So the key message here is sponsors must anticipate these risks and plan for them, and also document the mitigation strategies. We all know that the clinical trials can last for months or sometimes years, and technology might change during this period. Changes like OS updates, security patches, DHT software and hardware changes, or complete discontinuation of the DHTs might occur. So when such changes occur, it is important to document everything, like what changed, when it changed, which participants were affected, and what is the reason for the change. So on, all these have to be documented. And it is also important to verify if the device is still fit for purpose by comparing the data before and after the update. And if there is a change in the measurements, validation may be required and compare the measurements with the reference standards to make sure it is validated even after the update has taken place. And sensitivity analysis may also be needed to assess the impact on the trial outcome, which is followed by any change control procedures as appropriate. So the key takeaway here is DHT updates may be inevitable. However, proper planning and rigorous change controls and thorough documentation can still help maintain data integrity. So when signing informed consent in DHT-enabled clinical trials, the participants should understand the digital element in the clinical trial. For example, as we have just saw, these DHTs might have some risks, like clinical and privacy-related risks. So the participant must be aware of these risks and the mitigation measures that are in place. And they should know what this device is collecting and how long and how it will be used and who will have access to this data and for how long. Are there any security measures that are available? And are there any costs for the participants? And what are the procedures in case there is any severe symptom and who they can contact? So the bottom line here is participants must truly understand DHT-related privacy, data sharing, and technology risks before they sign the informed consent. So to bring everything together, when we are selecting a DHT, it is important to make sure that is fit for purpose, keeping in mind these three elements: the clinical trial population, technical specifications, and design factors. So in order to accommodate diverse participants in the clinical trial who may have different education levels, cognitive abilities, or technical aptitude, having features like multi-language support and plain language instructions, and having accessibility features like larger font size, screen readers, and voice input compatibility, all these features might help the participants with different needs. And when there is a caregiver involved in the clinical trials where you are studying in a pediatric population or cognitively impaired population, it should be designed as it required for a proxy use. And coming to the technical specifications, validation is very important. Again, it is important to confirm the accuracy of for the intended measurement, and the device should be compatible with different OS, and it should have adequate storage capacity. And when it comes to hardware specifications, it is important to verify the sensor performance and the battery life sufficient to minimize charging burden. So with these features, we are trying to make sure the participant don't feel burdensome. We are making their life easy, and their experience should be satisfactory. And again, the same thing goes for design factors. The interface should be user-friendly with clear instructions and larger font size, and with multi-language support. And they should also have flexibility of offline functionality if the participant don't have internet connectivity, with auto-sync capability where the data can be transferred when there is an internet connection. So the goal here is selecting a DHT that is fit for purpose for the study population and also supports the data integrity. So what are PROs and EPROs? A patient-reported outcome is any report of the status of a patient's health condition that comes directly from the patient without interpretation by a clinician or anyone else. These are direct patient-centered perspectives on treatment effects. Usually, PROs capture the health aspects that are not usually measured by laboratory tests or physical examinations. such as pain levels, sleep quality, ability to perform certain activity, or quality of life, or symptom severity or duration. And these PROs have been increasingly used to inform regulatory decision making. So what are ePROs? ePROs are the PROs which are collected using digital health technologies, for example, electronic diaries. These have become the cornerstone of modern patient-centric trial design. These ePROs enable us to collect data remotely in real time, and they improve data quality and accuracy, and also reduces patient burden by lowering the number of clinic visits needed. So this brings us to our use case, electronic diaries. Electronic diaries are nothing but the paper form of diaries, electronic form of paper diaries that the participant can use to record information during the clinical trials. So in vaccine clinical trials, the e-diaries are mainly used to collect solicited adverse reaction, including local and systemic events. They may also be used to collect unsolicited adverse reactions. Typically, the participants would report these adverse events for 7 to 14 days post-vaccination within a protocol-defined time interval. So why these e-diaries matter for vaccine safety? So usually vaccines are given to healthy population, including children. So we have very low tolerance for any unidentified safety events. With the help of these e-diaries, as we can collect real-time data, we can identify any safety signals and respond towards them in a timely manner. So why move from paper to electronic diaries? As you may know, there are several drawbacks from paper diaries. One of the main drawbacks is the recall bias. There is something called the parking lot effect, where the participant tries to fill the paper diaries sitting in a parking lot just before their clinic visit, trying to recollect or recall how they felt several days or weeks ago. So this kind of data will lead to inaccurate or missing data and will undermine the validity of the safety assessments. So in the next few slides, we'll see how using electronic diaries might overcome some of these drawbacks and what it takes for us to implement them correctly. So here you can see the workflows of paper diary and electronic diary. So with the paper diary, the participants will be given a blank booklet or a paper diary at their first visit. And then the participant starts to fill their symptoms by hand. And the investigators will review this data periodically when the participant goes back for a clinic visit. And then the study staff will enter this data into a database manually, which is a time-consuming and error-prone process. And at the end, there is no assurance that the data that the participant entered is accurate, and they cannot go back to correct the compliance rates. And when we look at the workflow of the e-diary, the participants will be set up a digital platform at their enrollment visit. And ideally, they will be trained during this enrollment visit on how to enter the data on the e-diary platform. And then the participant starts to enter the data in real time, and they will also be receiving automated reminders and alerts. So since the diary closes within a reporting, after the reporting window, they will have to enter the data in real time, which will avoid any recall bias or inaccurate data. And as they fill in the information, the data is instantly and securely stored in an electronic database. And the study staff will have access to this data real time, so they can monitor the compliance rates and for any data quality issues in real time. And also the e-diaries, these platforms can deliver the Alcoa Plus principles with the user authentication and full audit trail. The data collected with these e-diaries is attributable and original. And since we are collecting the data in real time and with time and date stamps, which will prevent backward filling, we are having real-time data. And with built-in validation checks, the data is accurate and complete. And the data that is collected from these e-diaries is usually stored in a cloud storage, and it is available and enduring. So all the features of these e-diaries will help us adhere to Alcoa Plus principles. And the main advantage of the e-diary over paper diary is having a date and timestamp. With all that said, e-diaries are designed to meet these principles only when they are implemented with appropriate controls and monitoring. So not all e-diary platforms are the same. So when we are building the platform, it is important to make sure that the platform is user-friendly and there are adequate comprehensive support systems within the platform. So when we look at the user-friendly design, again, the interface should be intuitive and they should have accessibility features, and it should have offline functionality and auto-sync capabilities for participants who live in the area of limited internet connectivity. And the participants should be able to customize the reminders when they can receive the reminders, and they should be able to visually see their progress, and they should have adjustable settings, because if the participant feels they are in control of their experience, the chances are high that they will be in compliance with filling these e-diaries. With any other technology, there could be some technical issues. However, with in-app tutorials and troubleshooting guides and responsive help desk, they should be guided to avoid any missing data. And with automated monitoring system, the staff can identify non-compliant participants, or if there are any severe events, and they can react in a timely manner. So the key message here is user acceptance is as important as technical accuracy. The best DHT is the one that the participants will actually use consistently and correctly. So even a best e-diary platform can fail if the participants and the staff are not trained well enough. So it is very important to train the participants during their enrollment visit. They should be able to fill the e-diary with the help of the study staff at their enrollment visit, and they should also understand the severity grading of the local and solicited events. And they should understand how to respond to the alerts and who to contact. And training of the study staff is equally important. They should be trained on how to set up the accounts, participants with the e-diaries, they are more convenient and less burdensome with reduced clinic visits, and they can get real-time feedback and engaging interface. And for investigators, they have real-time access to the data, and they can have access to the compliance rates in real time, and there is no manual entering of the data into the database. And for sponsors, improved data quality means stronger regulatory submissions. They can have data availability. They can have faster data availability, and they can. So with that said, these benefits from e-diaries can be attained with careful planning, proper implementation, comprehensive training, and active monitoring. So these technologies enables all these benefits, but the staff or the team has to deliver it. So in conclusion, DHTs have revolutionized our ability to remotely obtain and analyze clinically relevant data, transforming clinical research. Fit-for-purpose validation is critical, ensuring that the DHT accurately measures the intended event in the target population. And successful implementation requires careful planning, participant training, and ongoing monitoring throughout the life cycle of the study. And when properly implemented, these DHTs can improve trial efficiency and enhance patient guidance documents, which are the foundational documents for
No? No question from the audience? Okay, are there any questions from online?
Yes.
I'm sorry.
The first question is about validation. How does one complete the usability validation, or there are standards as how to conduct these validations? For example, how many subjects need to confirm that they are comfortable using the device?
So the question is, how do we conduct validation? So it depends on what you are looking at. So it could be something like you compare the measurements that you got from the DHT and compare it with the standard procedure. For example, maybe you can compare the step count from a DHT with what you observed. Or it could be you compare with the standards.
The second one.
Okay.
The second online question is about the IND submission. If e-diary information, such as screenshots of the interface, are needed for IND submission, if a sponsor plans to an e-diary in a trial included with an initial IND, but does not have the e-diary vendor or other operational details finalized yet, should they delay IND submission, or is there a way around this?
I'm sorry, could you repeat the second part of the question?
The second part. So if a sponsor plans to use a e-diary in a trial, included with an initial IND, but does not have the e-diary vendor or other operational details finalized yet, should they delay IND submission, or is there a way around this?
Okay, so the question is, what if we don't have a vendor at the beginning of the END submission? Should we wait? So typically, when there is an IND submission, we ask for e-diary screenshots of the interface and also the questions that you are asking the participants, because we want to make sure that the e-diary is used as intended as per the protocol. And we want to make sure that the questions you are asking to the participants and the options you give for each question, in case you're asking for the adverse events, the grading scales align with the protocol grading scales. So we typically require that data during the submission.
Are there any more online questions?
Yes. Which one? Okay. One more. When should the FDA be contacted in the development of medical products where DHT is used for data collection?
Yeah, that is a very important question. As you have been hearing from all the speakers this morning, it is important that you come to us very early as possible, to make sure the expectations of both the parties align. And if you need any specific guidance on how to use the DHTs in your clinical trials, there are several programs with which you can consult FDA. For example, the Q submission program, and there are several other programs. You could look into it in the DHT guidance document, or you could also go to the Digital Health Center for Excellence webpage. They have a lot of links for all the programs, and they have frequently asked questions section where you can get answers for most of your questions related to the DHTs.
One more online question. How should updates be handled when participants use their own device for data collection?
Yeah. So when you are using bring your own device, I think first you have to look into advantages and disadvantages of using that approach. When the participant allowed to use their own device, which they are already familiar with, it would be easy for them. And also they can avoid carrying another device along with their personal device. However, when a specialized device is used for a specific purpose, for example, a device that is looking into a measurement, and the algorithm is measuring a specific clinical endpoint, I think bring your own device may not be an appropriate approach in that case. So if it is a general platform like an e-diary app on your mobile device, I think it should be okay. And in that case, we have to make sure the updates do not interfere with how the data is collected. And if necessary, it needs to be validated to make sure there is no impact on the trial outcome. Thank you.
Thank you. Thank you, Dr. Nukala, for a wonderful presentation. So now we will move to session 4. Modernize the adverse events reporting. So as everyone knows, the modernization about the adverse event reporting means replacing outdated paper-based process with smarter real-time reporting technologies. So in practice, this will involve several key shifts. For example, real-time reporting. So critical safety information is available immediately, not days or weeks later. And the artificial intelligence and the automated system can process data faster and more accurately. And the standardized data formats can also enable smooth communication across systems and the regulatory bodies. So together, these shifts will improve data quality, boost efficiency, and ultimately strengthen the ability to protect patients and respond to safety signals. So for this important topic, I have the pleasure to introduce to you our session speaker, Dr. Luis Santana-Quintero, and he will be talking about this topic and also outline the biologics effectiveness and the safety innovative methods program. And Dr. Santana-Quintero has served with FDA CBER since 2012, leading the High Performance Integrated Virtual Environment, HIVE, bioinformatics groups, and key initiatives in computational biology, AI, and machine learning. He specialized in immunoinformatics, transcriptomics, megagenomics, and the application of AI to analyze large biological data sets. So prior to CBER, he worked as a postdoc fellow at George Mason University, where he utilized computational methods to study the effect of complex operators in evolutionary algorithms. He holds the Master of Science and PhD degrees in computer science from the Center for Research and Advanced Studies in Mexico. Without further ado, let's welcome Dr. Santana-Quintero.
Thank you. Thank you, everyone. Thank you for inviting me. Thank you for giving me the opportunity to present, to share with all of you the advances that we are doing at FDA, in specific at CBER, and how we are trying to modernize adverse event reporting for CBER-regulated products. So before I begin, I would like to acknowledge my co-authors like Jessica Zhu and Mark Walderhog, whose contributions are essential to the work I will be presenting on behalf of the team today. So yes, next slide. I'm going to start with the outline. Outline is, I will present some background information. I'll guide you through the study methods that we applied. I'll share with you some of the results approaches, including the use of AI to detect and validate adverse events associated with vaccines and biologics. Also, you will be able to explain the importance of collaboration between healthcare systems and the FDA, with the ultimate goal to ensure that the safety of vaccines and biologics. BEST IM program, where BEST IM stands for Biologics Effectiveness and Safety, Innovative Methods, and this work has been done in collaboration with the MITRE Corporation, that they help us with a large amount of the analysis that you will see today. So before starting on part of the background, I would like to give you an overview of the landscape of the products that we, in the Center for Biologics, we are regulating. You can see that there are products in the color boxes, where there are vaccines, gene therapies, blood products, and other therapeutics. And in gray, in the gray boxes, you see the exposure of these products in real-world healthcare, like pharmacies, emergency departments, universities, hospitals. And this is the BEST IM goal is to modernize the safety surveillance of vaccines, blood, and biologic therapeutics used to care for patients, again, with the ultimate goal to identify and evaluate adverse events tied to those exposures. currently, the advanced event reporting, it's a manual process. That relies largely on manual methods. You will see on the next page that these methods are based on the vaccine adverse event reporting system form, where there are many required and optional fields. Required fields are highlighted in yellow. In yellow, you'll see that we are required to get patient demographics, vaccine details, timing, clinical course, and outcomes. Completing this form typically means pulling information from different parts of the medical record and re-entering it here. Often they are still using paper or fax to do this. So looking at the challenges that this presents on the current methods that is existing with paper and fax processes creates burden. We can get under or over reporting and manual detection, individual flagging of these potential adverse events, manual validation, where there's time intensive for reviewers to be able to go through all the different sources to analyze and validate them. Potential adverse events are not always communicated, and then manual reporting, right? Data re-entry to report externally, lack of granularity in report evidence. So these are the challenges and the best IM opportunities that uses innovative methods to reduce this burden. It tries to increase the quantity and the quality of EA reports by automating this detection. How? By using new methods. I will show some of them using AI that can help us to score some potential cases. Semi-automated validation, where we can also use technologies to flag and prioritize these cases that are sent for review. And semi-automated reporting that helps to auto-populate the evidence and generate evidence based on the individual case safety reports. So all these are opportunities for best IM to improve the processes. And I will, this is an overview of the best IM platform, how it leverages the standards like the fast healthcare interoperability resources standards to enable the health systems to transfer electronic healthcare records data via the health information exchange and the qualified health information network infrastructure. So it comes to the best IM platform that resides within FDA, and within the platform, reviewers are able to identify potential adverse events, generate standardized reports, and provide a rapid notification and response. Ultimately, the ultimate goal is to use these standards and trusted networks to improve vaccine and biologic safety through the real-world data, faster insights, and of course, better outcomes. I will make a parenthesis here to mention that the legal framework that exists for data sharing that allows FDA to use electronic health records for surveillance activities. And this is through the data use agreement, or DURSA, that allows the exchange of information among e-health exchange participants for many different purposes, like including treatment, payment, and healthcare operations defined by HIPAA, innovative payment models, public health under applicable laws, meaningful use of certified EHR technology, and ultimately individual access, like patient access to their own health information. On the right, you see the letter that allows FDA to use this data is part of the Sentinel initiative that allows us and the best IM to use the data for surveillance. I'm gonna go with you with the best IM roadmap, how we got here. It started in 2019, where there was a prototype to build an EHR data pipeline. We started with a proof of concept exchange architecture, and in 2022, 11 health systems participated in a pilot to send some adverse event data to FDA using the FHIR service. So FHIR data. So currently we are looking to expand to make a mature and expand this adverse event validation by including more health systems into this network. Now I'm going to talk about the study methods of what FDA is doing with the data that we are getting from the health systems, right? So FDA partner with two different entities for two different studies. One of them is myocarditis, and look at phenotypes like myocarditis and pericarditis for vaccine exposure, specific for mRNA vaccine. And the second study is to look at another phenotype, the hypogammaglobulinemia. That it's derived from it could be derived from CAR T cell therapy that is used for cancer treatment. I'm going to talk only about the first study, even though I'm going to mention that the other one also exists. So I'm going to share results about the first one. I may not have time to talk about the second one. So to focus on the first one, for myocarditis and pericarditis, as I mentioned, we are looking for exposure of mRNA vaccines, specific for COVID-19 and I think RSV patients. The setting is we partner with two large healthcare systems that provided data via the eHealth Exchange using HL7 FHIR. Outcome measures, we are interested in myocarditis or pericarditis within 40 days after mRNA exposure. We are following the case definitions by the Brighton Collaborative. I'm going to explain a little bit more about that, how that we incorporate that into our analysis, and then how we make these comparisons with the AI technologies based on LLMs, and compare them with the clinicians' adjudications that we consider them as gold standards. So performance statistics included the Matthews Correlation Coefficient, MCC, and F1 score that are very good metrics for results when you have a binomial classification, like in this case. You will see what I'm talking about. Important to mention that in this study, we are not looking at we are not trying to answer the question of was the mRNA vaccine the cause of the myocarditis or pericarditis. We are just trying to identify if patients have these phenotypes or not the cause of it. So going to more in depth of the methodology that we are using is we are using the FHIR EHR data that is coming through the eHealth Exchange. We are textifying this data. We're using large language models to identify and adjudicate of these adverse events related to mRNA vaccines. We are using Llama-3 with unstructured data texts that are the clinical notes and structured data that are ICD-10 codes to provide an answer. We are using the Brighton Collaborative Adverse Event Definitions that helps us to create a foundation for the questions that we are asking for clinicians and AI-based methods on large language models to be able to to compare at the end and validate the automated workflow with them. Very important to mention is that we use 3 clinician adjudicators. We establish those as a gold standard data set to validate all our results. So yeah, this is another workflow of the big picture of how we are bringing data to do the analysis. As you can see, number 1, healthcare systems and FHIR identifiers patients of interest to FDA. We get these patient identifiers and we ask for the data via the eHealth Exchange. Routes them to the best platform, we get it inside FDA, and then we use it for both. As I mentioned, for our automated workflow using LLMs, and clinicians also make their determinations that we use to compare. At the end, we compare these methodologies, and you will see the results of this comparison later. In specific, I'll try to focus on the LLM, how we use the LLM. As you know, we are using resources from structured and unstructured data. We are asking LLMs different questions. We are asking them for a yes or no answer, a confidence, and a rationale of why they made that determination. Important to mention that during this process, we observed that refining the prompts was critical to get more accurate results. And you will see later how this improvement of the prompts help us to get more accurate results. We use two techniques that are, one is a chain of reasoning, and the other one is a few shot learning that we incorporated into the prompts. Bright-on collaborative criteria. These are the same criteria that we're using to ask clinicians to adjudicate the phenotypes to the patients, and the same language we're using for the LLM. In this case, there are many different criteria that they are looking for, like cardiac symptoms, non-specific symptoms, biomarkers, ECG abnormalities, etc. So the same the same workflow we are using for both. We develop SOPs to guide the clinician review of these phenotypes to try to be consistent on how they are adjudicating this into the data that we're receiving. For the study one that I mentioned before, the participants that we collaborated with are Cedars Sinai Medical Center in LA and Johns Hopkins Health System, where they were able to provide information, EHR data for 70 patients with myocarditis and pericarditis. Once we got this data, we divided in two sets, one of them with 20 patients, where we used them to kind of improve our models using LLMs, improving the prompts, and on the test set, we use the rest, like 50 patients, to evaluate the performance of these results. Again, the metrics that we are using are MCC, Matthews Correlation Coefficient, and F1 scores. Beforehand, we decided that an excellent performance will be an MCC greater than 0.8 and an F1 score greater than 0.5. Now I'm gonna go to the results for the myocarditis. Only I'm not gonna have time to show you the results for pericarditis. Improving the accuracy. Also a different level of comparison, and when we compare them, you see the results that it increased the agreement between the LLM-based methods and the clinicians. So you can see the MCC numbers are higher, in particular for the first cardiac and non-specific symptoms, echocardiogram abnormalities, and cardiac magnetic resonance abnormalities. Those numbers are higher than the other ones, like the cardiac biomarkers and ECG abnormalities. We see a less level of agreement. It just tells us that LLMs are not as good as identifying these biomarkers from the data as the clinicians. But when you group them and then you make a final adjudication, you will see that it compensates with the final results. On here, it's the final outcome determinations of LLM versus clinicians. You see they agree mostly in all the patients that we had. Only 2 of them the LLM marks them as definite, having myocarditis, when clinicians agree that those two patients were probably they had myocarditis. On the final performance metrics for the final determination of just a simple answer is like, did the patient have myocarditis? Yes or no? We see that they agreed in all of the patients. For the test set, we see the score of 1. So this tells us that the LLM was able to agree with humans, with clinicians, by following the same rules that we defined based on the definition of the cases. So this is just a distribution, just to share with you the distribution that we are getting for the patients. You see that in this case, most of the patients had myocarditis. Only one patient didn't have myocarditis. It's an imbalanced data set, but it was expected because from the beginning, we asked for patients with myocarditis or pericarditis. So it was just a way for us to confirm that the models are working, even though in that single patient, we were able to determine that it didn't have myocarditis. So in summary, for the large language model assessment, we see that how well did the LLM do compared to humans. For the performance of the phenotype components, we say that it was an excellent in determining presence of symptoms and echocardiogram abnormalities. It was not as good to be able to identify specific biomarkers, especially when they have numbers like thresholds, and identifying numbers and determining if it was above or below a threshold, it was not as good. For the outcome determination, it was excellent when it was using the clinical decision support engines. Again, ongoing optimization helped us to improve the performance of the LLM. And then for causality determination, as I mentioned before, it was not the focus of this study. And probably for future, we can make causality and severity determinations, even though it may require some adjustment of the prompt and the optimization logic that we use for those specific cases. About the challenges that we found, we believe that addressing these challenges is essential to enable high quality, scalable, and generalized real world evidence for public health. The challenges that we found is more participation from health system is needed, more health systems connected to the network. It will help us, FDA, to gather more information about these adverse events, about the EHR data, and bring them into FDA. Still, it's a voluntary participation. We need to be aware of technological advancement. Right now, we are using LLMs. We need to keep up and start looking at new methods to be able to adjudicate those by using AI. And then for our current validation study design, we should be including more negative cases to add and help resolve this problem with the imbalanced data set that we saw for the myocarditis. So in conclusion, we demonstrated that the use of AI, it's a good tool to help understand text, process data, enable automation, and be able to compute on these AE phenotypes, able to improve the precision also as well. Important to mention that this has to be evaluated by expert reviewers, clinicians. We cannot just use AI for now without human intervention, without the human in the loop. But we can use it to generate evidence and detect those signals, but they need to be confirmed by clinicians at this point. And again, the ultimate goal will be to assess risk and inform decisions, improve outcomes, and protect public health. In terms of the next steps, we believe, again, expanding this network with more participants. It's very important. Have this human-machine teaming up. We should not, again, rely only on the machine. For now, we should always have these reviewers taking a look and making the final determinations. It helps with the protocol standardization, trying to compute adverse events like phenotypes in an accelerated way. We can expand our studies to new phenotypes, to test and validate them, and then finally to share our results. We need to publish the results and share them with everyone else. And this is an acknowledgement to the MITRE team that without them, this work wouldn't be possible. Collaborators, leadership, and yep, I'm happy to answer any questions.
Thank you, Dr. Santana-Quintero, for a great presentation. So now we open for Q&A for this session. Are there any questions from the audience in the room? Please come to the mic if you have any questions you would like to ask the speaker.
Thank you very much for the presentation. You mentioned that the results showed low performance in identifying cardiac markers.
Yep.
But wouldn't there just, I mean, I would suppose you could teach the LLM and retrain it on new parameters that were more exact.
Correct. Yeah.
This could be in a future.
Correct.
In a future study.
This was the work that we did, I believe, last year, in which we observed these results. The current methods, as you know, they have advanced tremendously, especially with the new technologies like agentic AI systems that allows to have more accurate results, especially on these cases where you're trying to extract numbers from specific text in the thresholds, right? So it's like, what is the value that I see on this report versus the value that should be the threshold? And when we ask an LLM these questions, sometimes LLMs are not very good at answering that. With the use of agents, it improves accuracy because agents have the capability of create code to compare and get an accurate answer. So you do that, you improve the accuracy, and you resolve these issues that we found about extracting biomarkers specifically from the EHR. Yeah.
Yes. And then second part to my question is you had shown a slide showed a CAR-T case study. What was the patient population?
Do we know?
I would assume it was...
For the second study.
It was multiple health information exchange data.
But we don't have the number of that population.
I was just curious because CAR-T is typically used in very specific cancers right now and is expanding to other areas. I was just curious about that. And will you be able to share those slides if you're not able to present the material today?
Yep.
Okay, very good. Thank you. Thank you.
So we are a question from the audience in the room. So are there any questions from the audience online?
Yeah, the first question is, what are the main advantages of automating adverse event reporting?
I would say the main advantage is to improve the patient, like the safety of the patients by automatically processing and getting data related to adverse events, and they can easily be adjudicated by FDA, by expert reviewers. The Best IM platform offers that capability that accelerates, it helps to accelerate by using these new technologies that I just described. So I think that will be the main goal for that.
The second question, can you share any information about the study for the CAR therapy that was particularly relevant for challenges in AE detection for CAR therapies, and that CAR indication in particular?
didn't hear.
It was basically the same question that I asked.
Sorry.
So like, what was the patient population and share the study?
Yeah, we don't have the numbers for the second study for the patient population. For the first one, yeah, you saw the patient population of 70.
So here is more about the challenges in AE detection. When you do that study for the CAR therapies.
I couldn't hear the question. I think the sound comes. Sorry.
The challenge.
Oh. I don't have any comment on that.
Okay, let's go to.
Similar to current, this past one talked about, you have to have agreement between the large language model and the clinical reviewers. So it's a little more complicated because it's a different set of symptoms and signs. So it's a learning experience in terms of how.
Okay, yeah. So basically, it's similar methodology, different prompts that we are using, right? Different case definitions for different phenotype. So that would be the main difference, even though the methods are very similar. Important that we should be including the clinicians' information, how they are adjudicating. That's why also we selected 3, so we can resolve any potential discrepancies among them. Yep.
The question is, what are the standards that allow data transfer between the health systems and the FDA within the platform?
Yeah, those standards are the FHIR data, the FHIR standards using the HL7 with the eHealth Exchange network to bring those into these standards so we can use them properly.
Another question is that how will the LLM prompts improved in the study and the difference of optimized set and the test set?
We mentioned that we had 2 different ways that we improved the prompts. One that was the use of reasoning, and the other one to use chain of reasoning and the few shot, right? When we incorporated some cases into the prompt that helps the LLM to provide better answers. And the difference between the optimization and the test set, where that optimization, that's the set that we use to improve our prompts without using data from the test set. Once we have these final prompts defined, we run the algorithm on this test set, and that's when we ended up getting those final results that we compare with the gold standard we ended up using. Mm-hmm.
So are there any more questions from the online? No? So should we prepare the questions?
Okay, we can finish and go back at 3:05.
We can finish it? Oh, okay. Thank you. Thank you, everyone. And thank you for both speakers for great presentation. And we will take, well, now it's not 15 minutes, it's more than that. So the next section, section 5 and the section 6 will start at 3:05. Thank you.
Thank you. Everything for the introduction was very nice. Thank you.
Thank you for coming. Thank you for you did it. Yes. Thank you.
Oh, that's a good question, right? Basically, the context. Yeah, so I mean, it's there's a limited context of the use of the LLMs. So we have methods to truncate the data. They're not perfect, but it would allow us to truncate data based on the able to forcibly fit into into the context window that we have, especially for all LLMs like Llama, over LLMs.
Are we online? Click it. Okay. It sound good? Everyone can hear me? Okay, perfect. So thank you for all of the attendees to keep up with this amazing talks throughout the day. So we have 2 more sessions to go before we have some closing remarks and make it to the end of this day. These sessions are going to be a little bit different than the previous one that you have seen. They're from the Office of Blood Review and Research. So our first speaker is Dr. Wendy Paul. She is the director of the Division of Blood Components and Devices, DBCD, and in the Office of Blood Research and Review here in CBER. Okay, so she has over 15 years of clinical experience in blood banking and transfusion medicine and 12 years of federal regulatory experience here at CBER. Dr. Paul joined the FDA in 2014 as a medical officer in CBER's Office of Biostatistics and Epidemiology. What used to be OBE, now it's OBPV. We had 2 speakers in session 1 and 2 that were from OBPV, right? And then she brings a wealth of analytical and administrative skills. In 2016, she joined OBRR as Deputy Director of DBCD and accepted the position of Director in 2026, so just recently, right? Overseeing the regulatory work of about 50 scientific and clinical staff. Prior to joining FDA, Dr. Paul held an academic appointment as an assistant professor of pediatrics and pathology at George Washington University School of Medicine, while serving clinically as medical director, point of care and satellite testing, associate medical director, transfusion medicine, and attending physician at Children's National Medical Center in Washington, D.C. So Dr. Paul has completed her undergraduate education at the University of Colorado, Denver, and received her medical degree from University of Maryland, Baltimore. Yes, so she completed an internal medicine internship at the Washington Hospital Center and residency training in anatomic and clinical pathology at Howard University Hospital in Washington, D.C. So following residency, she completed 2 fellowships, one in diagnostic immunology at the Johns Hopkins Medical Institute, and another one at the Blood Banking Transfusion Medicine at the University of Virginia, Charlottesville. Yeah. So without further ado, I'll welcome Dr. Wendy Paul. Thank you.
Good afternoon, and thank you for staying present to this late in the day. Very glad that you're here. I am Wendy Paul. I'm the director of the Division of Blood Components and Devices, also known as DBCD. I am very happy for this opportunity to present to you today and to share with you our regulatory perspective on novel blood components and related products. And I was here earlier in the day and I heard Dr. Scott from OBPV present, and he said next time he'll make a shorter bio. Note to self, just a little awkward, but it's interesting. As much as you do, everything you do, I think, has meaning and purpose. But as much as you do outside of these walls, you bring it with you. But it's a whole new world in here. I'm very happy to be on this side of the wall today. So we'll get started. I would also, before I start, like to acknowledge that one of my former FDA colleagues was very instrumental and developed most of the slides in this presentation today. And he was preparing for this conference last year that was not held. So I would like to acknowledge Dr. Carlos Villa, who a former FDA colleague, for preparation of these slides. So let's get started. So the overview of today's presentation, we are going to discuss FDA's regulatory framework for blood and blood components. We are going to define novel blood components as biologics. They can also be output of medical devices, and they can be drugs. We're going to review the applicable regulatory pathways, and finally describe product life cycle for biological products. I hope you'll find this information is useful. Just to give you an overview, you've been here all day today and maybe jumping in between tracks, but this is CBER, Center for Biologics Evaluation and Research Review, and OBRR, the Office of Blood Research and Review. And I just want to talk to you about, our activities. For OBRR, my office, we're primarily focused on product regulation. We want to make sure that the products being brought to the American public are safe and effective. But we also do things that some people don't know about. We participate in blood establishment registrations. We conduct inspections for compliance and enforcement. We conduct post-market product surveillance, and that includes things like post-market safety studies, review and adjudication, internal adjudication of fatality reports related to blood and blood components, medical device reports, MDRs. And we also very, very involved in the development of rules and guidances that you use to develop your products and get them out there on the market. And the final point is that in OBR, and investigational mode. So I'm going to start by just walking you briefly through the legal framework for FDA and our regulation of biologics. The regulatory framework for everything that we do in OBRR is supported by the Public Health Service Act, also known as PHS, and this is involved in the licensure of biologic products and the control of communicable diseases. And it's also supported by the Federal Food, Drug, and Cosmetic Act, also known as the FD&C Act. And this is to ensure that good manufacturing practices are put in place for drugs, medical devices, and blood. Biologics also are subject to some of the definition of drugs. So they're subject to certain provisions within the FD&C Act, which include GMPs, labeling, and ensuring that there's no misbranding or adulteration. In addition, we publish, I'm sorry, I'm getting a little ahead of myself here. So we also then, from the statutory foundation, develop regulations that implement the PHS and the FD&C Act. And specifically, our regulations are found in Title 21 Code of Federal Regulations, which we call 21 CFR. And those regulations for blood and blood products are found in sections 600 through 680. These regulations do have the force of law. And we additionally publish guidance documents, which we think help the public interpret what is our current thinking about the regulations. However, our guidances, you should know, do not bind FDA or the public. And we also want people to be aware that while our regulatory framework is very structured and very well defined, alternative approaches can be used if they satisfy the requirements of the applicable statutes and regulations. So in other words, we are willing and sometimes able to find other ways to get to what the regs intended. The specific regulations for blood and blood components are found here. I'm not going to go through each section of 21 CFR Part 600s, but I just wanted to list where the specific regulations for blood and blood components are found, and to let you know that this is what we apply to our conventional blood and blood components. And since this talk is about novel blood and blood components, these regulations would also be the basis for any novel product that you are considering development for. Okay, I got my directions mixed up here. Okay, so in addition to the very well-defined statutory and regulatory framework that we have, we also, our activities are influenced by outside factors. And so one of the things that may influence our regulatory activities are responding to emerging and emergency public health issues or problems. We also, as we're developing or as we're interpreting our regulatory framework, we want to include the relevant input from relevant stakeholders. And that includes both internal at levels higher than OBRR and possibly CBER, and also our external stakeholders, which would be the industry and our public health partners. We also we try as much as possible to be responsive to those external concerns, but we stay within existing statutes and provisions. And then there's always the response that we respond to new statutory enactments that may affect us. So I'm going to quote another couple of people that I heard earlier today during the plenary session. Now I'm blanking on which center deputy said this, but he said, "Tell us your story." And it resonated with me, and it obviously resonated with some of the other speakers earlier today. And I just wanted to share that with you. And when I was preparing this talk, one of the tips for preparing a good talk was to tell a story. So this struck me. I know you've all heard the idiom, "the greatest thing since sliced bread." And I just kind of Googled it and did some digging into it. And it refers to a major advancement applied to modern technology or convenience products. And interestingly, prior to 1928, the idiom was, "the greatest thing since bread was wrapped." So the bread was wrapped, but it wasn't sliced. So that's what people used to say. The automated bread slicer was invented. The first prototype was invented in 1912, and due to a series of warehouse fires and different mishaps, the actual slicer didn't make it to the light of day until 1928, and that was when the first loaf of pre-sliced bread was sold in the U.S. I thought this was interesting and applicable because we meet with a lot of sponsors, and we see a lot of initial packages, and and we think it's good. People are convinced that what they're doing is an improvement over what exists. Sometimes it is. And if you don't believe it, how are you going to make us believe it? But we want you to tell your story. And so the story here is that good things take time. Sometimes they take time, but sometimes they are better than what existed before, or at least as good as. So we're going to talk now about novel blood products. And basically what I'm going to focus on is your questions and how you can think about new ways to slice the bread. Blood is blood, right? It is a human-derived medical product. A medical product of human origin is what they refer to it kind of officially. But there's always new ways to package it, to store it. Some of the things that we see kind of, not frequently, but we see kind of recurring themes are optimized storage and availability, reduction of risk. So, you worry about infectious disease risk, you worry about immunomodulatory effects of blood. So there's all these attempts at reducing risk. And then there is personalized therapies. And I'm not venturing into the cellular therapy world, but I'm really talking about how we went from transfusing whole blood to components. And now the pendulum is swinging back to a lot of consideration about whole blood, but it's really about giving the product that's needed in that particular situation. So because blood and blood components and blood-like products are all medical products of human origin, I would just like to say that trying to find a new way to slice the bread is complicated. My office director said that to me, and it was an aha moment. It's complicated. So here on this slide, I've just listed some of the novel products that we've seen. Because we are the Office of Blood, we see everything. We see the whole bloods. We see plasma products, platelet products, red cell related products. This slide just lists some of the novel plasma products, and these are all available for your interest in public domain, but we've seen freeze-dried plasma, spray-dried plasma, solvent detergent treated freeze-dried plasma. There are novel red blood cell products, and basically these are what have been referred to as artificial blood, which is kind of a popular pop culture terminology, but it really refers to an oxygen carrier and often derived from red cells. Hemoglobin-based are one of the classes. There were the perfluorocarbon oxygen carriers, but if you're familiar with this field, you'll know that it's been under development and under investigation for decades, and there have been products that have made progress. One of the challenges is designing trials that demonstrate both the safety and the efficacy. But again, there are some new players on the block now. There are the biosynthetic artificial red blood cells, or some of the perfusion solutions that are hemoglobin-based, not necessarily human, but hemoglobin-based perfusates. So again, it's complicated. And then novel platelet products. So we've seen cold stored platelets, we've seen cryopreserved platelets, we've seen some of the lyophilized platelets. And I'm gonna focus just today, because blood is so broad and there are so many different components within blood, and whole blood, it can be broken down. I'm going to focus on novel platelet products as sort of a test case that we're going to discuss on what to consider when bringing forward one of these products. So novel products require novel approaches. The specifics of our regulatory approach to any given product is very likely to depend on a product-specific assessment. And that would include, as our previous colleagues from OBPV would include, the benefits and the risks of the product. What is different about the product than the characteristics of the conventional product? Sponsors and stakeholders have questions on their specific product or their plans for development. We urge you to contact FDA. We want to hear from you. We want to talk to you. We want to hear your ideas and where you're going. And engagement with regulatory authorities throughout this process, and this is not a new point, everyone has made this point, is best done early so that you avoid mistakes or missteps that you might not have to take. So we're going to now dive into novel platelet products. And the remainder of my talk today will focus on our regulatory perspective, and we'll use this platelets as a test case. The principles presented here, though, can be applied to any of the other components. You just need to focus on the indication, the product characterization. So, surprisingly, novel products are already in the CFR. We just don't call them novel products. This I'm not going to read it, but this excerpt before you is from the Federal Register from 1974. And it discusses the regulatory perspective on the appropriate storage temperature for what we now refer to as conventional platelets. And these are medical products of human origin. And it says that they're stored at room temperature. You get a better post-transfusion survival time. However, platelets stored in the cold appear to be more potent during the initial stages of hemostasis, which is important in bleeding patients. So what temperature is appropriate and for how long? And that was a question that was being discussed as far back as the 1970s. And so the answer, obviously, the commissioner says that you can store the product at either temperature and utilize the processing, labeling, and distributing procedures that are consistent with each. So again, the bottom line here is it depends on the indication and other considerations. So this is where it gets a little more complicated. What are the storage bags? What mitigations are you going to use for bacterial risk reduction? Does the product after so many days demonstrate safety and effectiveness or efficacy? So this is just an overview of platelets intended for transfusion. They're usually collected, manufactured by blood establishments, and sometimes can be subjected to additional processing steps. These steps are usually carried out in the blood establishments that manufacture platelets and are relatively simple. But the regs do require that you use a cleared or approved medical device in accordance with their instructions for use. So some very basic examples include leukocyte reduction, irradiation, dividing the product into multiple aliquots, and pathogen reduction. Not relatively simple, but pathogen reduction. Let's start at the beginning. Blood establishment licensure is outside of the scope of what I want to talk about, but I do want to call attention to a very excellent webinar that was conducted a week ago today, actually, by the Blood and Plasma branch, which is one of the branches within the Division of Blood Components and Devices. And I've put the link here in this slide because the recorded session is available for your viewing. So that if you are a licensed establishment or seeking licensure, this webinar gave a textbook, a wealth of information on how to accomplish that. In addition, there are multiple platelet guidance documents. And here, this link in this slide is a link to the blood guidances on the CBER website. And it includes guidances such as how to collect platelets by automated methods, alternative procedures for the manufacture of cold stored platelets when conventional platelets are not available or their use is not practical. And then a very, very useful, and I feel like groundbreaking guidance was the bacterial risk control strategies for blood collection establishments. So there is a list of all of the current and active blood guidances at this website. So we've talked about collection of platelets, very basic at a very basic level. We've talked about licensure for facilities that collect platelets. Now we're going to talk a little bit about the device paradigm. So blood establishments that manufacture platelets, I've already said this, need to use cleared or approved devices in a manner consistent with their instructions for use. If your device is going to be used in the manufacture of a novel platelet product, we would have to then decide what class your device belongs to. And I just put this in just to introduce you to the fact that there are some class 1 all the way to class 3, with the highest risk device being considered a class 3. So the class to which a device is assigned determines what kind of regulatory submission you need to make. So a class 1 is an exempt device, and you don't usually have to do a pre-market notification. A class 2 would be some of the things like a leukocyte reduction filter, or some of our blood collection and storage systems, which are 510K and class 2 devices. And then you have the ones who have the highest risk and our class 3 devices and require submission of a pre-market approval application or a PMA. If a product differs from a conventional platelet and is what we're calling a novel platelet product, it may be subjected to further manufacturing processes that are much more complex. And this can result in a product that is sufficiently different than the conventional platelet would be. And therefore, we would regulate it as a novel biologic. And in most cases, this turns out to be a combination product. We expect that the scientific understanding of the effects on the platelets and the risks to the recipients might not necessarily, let me back up, I got ahead of myself. So we have a specific understanding of our conventional blood products, and we understand their effects, their clinical effects, and their risks to recipients. But the risks of the novel product, the modified product, may not be the same. So we expect that the sponsor would develop an understanding of those different effects and different risks. There are some products that are out there now being investigated that are synthetic or have semi-synthetic approaches, and they may be used to manufacture products that are not necessarily platelets or platelet-derived, but may mimic some of platelets' function. Generally, those are considered entirely new products and can either be a drug, a biologic, or a combination product. And the type of product that it is will be determined by what is the primary mode of action. And then the product would be assigned to the appropriate center and the appropriate review team, but with lots of collaboration and input from the experts in other centers if needed. So right now, all of the things that I'm talking about are platelets and platelet-like products are being regulated in the Office of Blood Research and Review. Biologic platelet-like products, so those derived of products of human origin, are generally regulated here in CBER. If it's a progenitor cell-derived product, it most likely would be regulated or reviewed by the Office of Therapeutic Products, where cell and gene therapies live. But it also might depend on what is the indication, what is the primary mode of action for the output. And then again, like I mentioned, CDER may be involved with input as needed. So we've talked about how to consider which regulatory pathway is appropriate, even how you might take a stab at what center to submit your initial contact to. But I'm going to talk now about how do you license a novel platelet-like product. So most biologics and drugs follow a life cycle to transition from the preclinical phase to the clinical phase, clinical investigation phase, and ultimately to approval. IND, which stands for Investigational New Drug, is defined and explained in 21 CFR Part 312. You should communicate with us early and throughout the process. But the details of each life cycle will be very product specific. So you can look at what's on clinicaltrials.gov and get an idea of what we've done with previous products and what we've expected or asked for. But each product has its own footprint, its own fingerprint, and your life cycle will depend on what you're trying to do, what you're trying to get. So let's talk a little bit about indications and usage of novel products. And again, this is focused on platelets. And it is expected that the indications and usages would overlap in the clinical settings where conventional platelets are transfused. But you know, in the world that we live in, we talk a lot about shortages, we talk about supply chain, we talk about pandemics and how to be prepared for the next big event. And so a lot of the product development is in preparation for when these conventional products that we understand are not available or may actually be the next best thing. And some of those settings are in hypoproliferative thrombocytopenia, in cardiac surgery where there is platelet dysfunction using conventional platelets, in other perioperative bleeding settings. And there's been a lot of conversation about traumatic hemorrhage and pre-hospital administration of blood and blood components. So the design of the clinical studies to support licensure or approval will depend on both the characteristics of your product and the indications being sought. Now, this is a great slide, and Dr. Villa put this one together. I'm not going to go over it in a lot of detail, but the title says what I'd like you to take away is that platelet transfusions are the standard of care in many settings where well-controlled studies are limited. And it just sort of highlights the complexity of developing a product to either replace or be equivalent to something that is already the standard of care, but that we don't have well-controlled data to compare with your data in your product in development. There are all of the biological and physiological attributes of platelets. There are the clinical considerations, like is this for bleeding or is it for prophylaxis to prevent bleeding? What is the patient population? The bottom half of this diagram is what are the product factors. So how long was it stored? How was it processed? And then because it's a product of human origin, there are all of the donor and recipient factors. ABO matching. Is the donor alloimmunized from previous pregnancies? That will determine how the recipient responds to the transfusion. And so then it really makes it complex when you try to develop outcomes of randomized controlled trials, which is what we're comfortable with receiving, right, to evaluate data, randomized controlled trials. So this kind of highlights the challenges of developing a new novel product. I hate to ask this, but where am I for time? 4 more minutes? 1 more. Oh my goodness. I didn't intend to talk this much. I apologize. I'm going to talk a little faster. This slide is self-evident, and if you want to take a picture of it, that's okay. But it just shows the schematic of the life cycle of a biologic product. The pre-IND phase is important for novel products because this is where you get that early interaction. You can go to the website and look up what is an interact meeting. You can ask for that before you even submit a request for a pre-IND meeting. And then you want to come in for a pre-IND meeting before you go through the exercise of designing a trial, submitting it, and we tell you we don't have enough single dose toxicity, or, you need to go back to the drawing board. So interact, then the pre-IND phase, all very important. And then this slide talks about what goes on during the clinical investigation phase. And there are phase 1 trials, which are focused mainly on safety. Phase 2, where we start to look at both safety and efficacy and determining the appropriate dose strategy. And then phase 3, which would then be powered to demonstrate efficacy and safety. Studies need to follow good clinical practice guidelines. And there's a link in that slide for the regs. I'm going to skip over what should be in your IND application. That's also on the website. But make your package as complete, tell your story. We exercise a team process for IND review, and every discipline that's needed to fully understand your developmental plans are represented on these teams. And we base our reviews on the best available science. Once an IND is submitted with the protocol and everything, you're either allowed to proceed, or based on safety concerns we have, we'll place your study on hold. And we iteratively work out what those deficiencies are and how you can satisfy them. Our goal is to help you do studies that are both useful and safe. I'm not going to talk about the BLA process because that comes after IND. If your product is being regulated as a biologic, your regulatory pathway will be a BLA. It depends on what the primary mode of action is. The primary mode of action I'll back up from that statement. But the BLA process, there is also a pre-BLA phase. So if you have a product that you want to bring in and the regulatory pathway is BLA, you can ask for a pre-BLA meeting and ask us questions about, is this the appropriate regulatory pathway? And the outcome of a BLA is different from an IND. You either get a complete response, meaning we found several deficiencies. It itemizes the deficiencies, but we outline them and tell you what you need to do to address them. And then the review clock stops while you gather that information and resubmit. Or if everything is in a row or after several back and forths with us, eventually you may get to an approval. And then the final thing that we do in this process is the post-licensure safety surveillance. So, and I mentioned this earlier, it's either done to further characterize the safety profile of your product, or we do that with a formalized study, or you can report if your product is a device through the FAIR system, which you've heard about earlier today. And you have to submit all serious and unexpected adverse events to us within 15 days. And that helps us to build the safety profile. There are certain special circumstances where we may require a phase 4 study, but some studies are commitments where it's an agreement between us, and some are a requirement, meaning that we can require you to do a post-market study. And if the conditions of the study aren't met, we can revoke the approval. But these are usually done to either demonstrate additional safety profile, or complete deferred pediatric studies, or address an unknown serious risk, but that didn't rise to the level of not approving the application, but additional information is needed. There are also expedited review pathways, and these are explained further in the link that I've shown here. And these are to help products that may be needed to meet an unmet medical need for the treatment of serious conditions. What it does, it doesn't compromise on the data and the information needed, but it gives you more frequent and more engagement with the agency prior to submitting your final package. So in summary, I've talked about how biologics, novel blood products are regulated as biologics. I've talked about how we, OBRR, like to engage with sponsors to support the development of your novel products. Stress that communication is key, and given some quick and high-level details about life cycle. So my closing thought is that your novel product may actually be the best thing since sliced bread, or if you're like me, it may be a baguette, which might be better than sliced bread. But talk to us, come in and exercise and utilize all the resources that we have available. And I'd like to thank you for your attention, and I apologize for running over.
We do have an online question. How does FDA determine whether a novel blood product is regulated as a biologic, a drug, or a combination product?
That's a good question. I'm sorry. Primary mode of action. Thank you, Ann. So we look at the primary mode of action of the output and determine whether it's a biologic, a drug, or a device. If you have questions, you can always submit a request for a meeting with us and give us some background and tell us what you're thinking and if whether or not we agree.
We have one quick one. What expedited review pathways are available for novel blood products, and when might a sponsor consider pursuing one?
Okay, so thank you. That was at the end of the presentation. So when you might consider requesting an expedited or consideration for an expedited pathway is when you have something that you consider to be novel and that it can potentially address an unmet need and for a serious condition. So if it checks those boxes, look at the different pathways that are available and see which one may meet your needs, and then contact our regulatory project managers to request a meeting with a set of questions that help us understand what you're thinking about. I want to thank my colleagues, Dr. Eder, Dr. Elo, who couldn't be here today, and to Dr. Villa for the use of some of the previously prepared slides, and to the organizing committee. Thank you very much.
Okay. Yeah. So our next speaker is Dr. Silvia Paoli. She is a biological reviewer in CBER, and she has extensive experience in both chemistry, manufacturing, and controls, also known as CMC, review and pharmacology toxicology review. So she received her PhD in chemistry from the University of Sao Paulo, Brazil, and has over 12 years of research experience in nanomaterial toxicology and blood product safety assessment, with 45 peer-reviewed publications. So during her 14 years at FDA, she has applied expertise across both CMC and pharmacology toxicology disciplines to evaluate the safety, quality, and manufacturing of biologics and medical devices. Dr. Paoli also served as deputy topic lead for the ICH Q3E guideline, which she will be talking about today, and supporting global harmonization of standards for extractables and leachables. So without further ado, I welcome to the stage Dr. Paoli. Thanks.
Well, thank you for the nice introduction. So again, my name is Silvia Paoli. I work for CBER as a biological reviewer. And I'm also the deputy topic leader for the ICH Q3, which is the topic of my presentation today. And so this new guideline represents a big development on how we approach this topic of extractables and leachables. How to advance these slides? Okay, so I would like to start with an introduction to the ICH. So the International Council for Harmonization of Technical Requirements for Pharmaceutical for use, Human Use, the ICH, is an initiative that brings together regulatory authorities from different countries and the industry representatives. The ICH guidelines are internationally harmonized scientific and regulatory standards that are developed with the goal to promote safe, effective, and high quality medicines. The FDA is a founding member of the ICH since the 1990. And the FDA implements ICH guidelines as FDA guidance documents. And those are the most clear and the most transparent signal to industry regarding the FDA expectations about extractables and leachables.
Sorry about that.
Mm-hmm.
Sorry about that.
Thank you.
Yeah, the clicker.
Okay. Okay, so the ICH Q3E is currently in draft stage. It's being released for public consultation last year. And currently the ICH expert work group is reviewing the feedback from all the stakeholders. The guideline is anticipated to be finalized between 2028 and 2029. And so after finalized, the ICH Q3 will represent FDA's current thinking on the topic, and therefore industry compliance will be expected for the applicable submissions. So the ICH has established a series of guidelines that address impurities in drug products that include the Q3A, ICH Q3A, that deals with impurities in drug substances, Q3B, that deals with impurities in drug products, Q3C covers residual solvents, Q3D inorganic impurities, and M7 that covers the mutagenic impurities. So there is a gap in here because none of these guidelines comprehensively address the extractables and leachables from contacting material with the drug products. So leachables are chemicals that migrate from contacting materials to the drug products while in contact with packaging or manufacturing equipment and medical devices. So the driver for this ICH development is not only because of this gap, but also because we see several advances in material engineering, a lot of device innovation, novel manufacturing methods, new therapeutic modalities, which include cell and gene therapy. And therefore, there is a need for forward-looking principles that are necessary for anticipating future trends in pharmaceutical manufacturing. Regarding the scope of the guideline, what is in scope? New drug products, including cell and gene therapies, drug device combinations, and it also applies to life cycle changes that affect the leachable profiles. The guidance is focused on organic leachables, since the inorganic leachables is mainly covered in the ICH Q3D. Several product categories are outside the scope of the guidances. For instance, impurities introduced in the drug product because due to impurities or adulterations, herbal crude products, clinical research stage, unless there is a high patient risk. Also, it excludes radiopharmaceuticals and the systems that are used in manufacturing and the storage of excipients. Excipients are dealt through impurity specifications and the other compendiums. So let me define the two terms that underpins the ICH Q3. Leachables are chemicals that migrate from contacting material from manufacturing packaging devices. Leachables studies are performed under manufacturing and labeled storage conditions. These studies show which chemicals will be in the drug product. Extractables, on the other hand, are chemicals that are intentionally extracted under laboratory extraction conditions, typically high temperature and harsh solvents. Extractables studies represent potential leachables, and it is very important to guide the targeted leachables studies. Importantly, both studies, extractables and leachables, are essential for a comprehensive safety assessment of leachables. The ICH-Q3E is based on a holistic framework built upon the ICH-Q9 quality risk management principles, which involve the 3 different steps. First, risk assessment, then risk control and life cycle management. Risk management encompasses hazard identification. That is when you identify extractables and leachables of concern. Risk analysis, that's when you estimate how risky those leachables are. And integrated risk evaluation, you determine if the risks are acceptable. Then in step 2, it's the risk control that encompasses risk reduction and risk assessment. In risk reduction, you take an action if there is a risk. And in risk acceptance, you justify why the risks are acceptable. And the life cycle management, you keep measuring the leachables profile until the discontinuation of the product. So to understand the risk regarding to leachables, it's important to understand the multifactorial process of the nature of the risk. So there are several aspects happening at the same time that contributes. From the pharmaceutical quality dimensions, you need to think about the potential interaction between the drug formulation and the equipment, manufacturing equipment, and the packaging. You also need to consider the physical, chemical, material property, including post-sterilization. You also consider the manufacturing process itself. You should look at the contact surface area and the temperature and the solvents used in the manufacturing. You consider the drug storage conditions, temperature and shelf life, and the drug formulation itself, because pH, solvents, surfactants, excipients, they all may increase or decrease the leaching property. In the safety dimension, it's important to look at the route of administration. For instance, if you have a solid drug that is stored in a plastic material, that scenario is less risky, generally speaking, is less risky than the same material is used to store a drug that is for use intravenously or for intrathecal, for instance, administration. You also consider the dosing of the drug, frequency and the maximum dose, and certainly the treatment duration. What is important to remember here is that the guideline, ICH-Q3, enables flexible, science-based approaches on how to tackle extractables and leachables. The risk assessment for extractables and leachables involves 3 distinct processes. Step 1 is hazard identification. Here, the question that you want to answer is, what are the potential leachables that could be in the drug product? Here, you identify extractables and leachables from direct contact and also indirect contact, such as secondary packaging, inks, glues, whatever is used in the product. You can also use prior knowledge with regarding to that material. And for that, you can use the literature, peer review literature, and also vendors' information. Step 2 is risk analysis. And the question here is that you need to answer is how much exposure a patient will be exposed to the leachables. So here is when you quantitate extractables and leachables, then you access the exposure, which is dependent on the route, in the dose, the frequency of dose, and the duration of the treatment. Step 3 is an integrated risk evaluation. And in this part is where you need to explain or justify why the exposure is acceptable. So you evaluate the impact of leachables on the quality, safety, and the efficacy of the drug products. And if everything goes well, you can consider the material qualified for its intended use. If there are risks identified, then risk mitigations and control strategies are required. Risk mitigation measures include changing the contacting materials or changing suppliers, washed components or manufacturing equipment, adding purification steps to decrease the level of leachables in the drug product. Control strategies could involve the establishment of acceptance criteria for leachables. And here also you implement relevant analytical procedures. And very importantly, you establish quality agreements with the vendor. That is important for life cycle quality controls. And such quality agreements should involve statutes that requires the vendors to tell you promptly if there is any change in chemical composition of the material or change in manufacturing changes. Mitigation measures and control strategy both needs to be verified, and that should be done through extractables and leachables studies and safety assessment of the leachables. So one aspect of the ICH Q3E is that it is applicable to life cycle management, and there are many changes that would require extractables and leachables re-evaluation. And the changes can be a change on it can be a new information, for instance, changes in the material suitability, or a new patient information with regard to a specific leachable. It is also formulation changes, such as composition or concentration of the drug product. Materials change. So if there is any change in material composition or suppliers change, manufacturing process, all that may trigger extractables and leachables re-evaluation. Drug manufacturing process change is also important. So changes in solvent temperature, pressure, also increase or decrease in batch size, and the cleaning, sterilization, validation. All those are aspects that can change the risk, the leachables profile, and therefore requires re-evaluation. Importantly, you also need to consider exposure to leachables change. For instance, changes in drug posology or treatment duration, a change in route of administration, and patient population. So the takeaway from here is that the Q3E establishes a life cycle basic expectations for extractables and leachables assessment and safety evaluation. The Q3E defines two types of extractables studies, a semi-quantitative and a quantitative one. The semi-quantitative extractable studies is a screening tool to identify which leachables can actually be present in the drug product as leachables. These studies are important to guide targeted quantitative extractables or leachables studies. A quantitative extractable study is when quantification of extractables above the analytical evaluation threshold should be performed. I'll be talking more about the analytical evaluation threshold in a couple of minutes. A quantitative extractable study requires confirming the identification of the compounds. And a well-done quantitative extractable studies can be supportive of material and components qualification in low-risk scenarios where you don't have to do anything else but a quantitative extractable studies. The key requirements for extractable studies are the following. First, the study should involve the use of multiple extraction solvents with varying pH and also polarity. It must use the worst case extraction conditions, so typically high temperature and long extraction time. And multiple analytical techniques complementary to each other should be used in order to evaluate organic chemicals that are volatile, semi-volatile, and non-volatile. So now let's turn to Leachables studies. These studies are very important because Leachables profile is what ultimately drives the safety evaluation and the material acceptability. Leachables studies are conducted in the final drug product, in its commercial presentation over its shelf life. And it includes also batch-to-batch variability assessment. Leachables studies can be targeted or it can use screening approaches. There is also a simulated Leachables studies, and these studies are performed when the drug product is too complex and it would interfere with the basic analytical methods. Another example would be, for instance, large volume parenterals, where the Leachables can be too diluted for identification and quantification. So in products like that, a simulated Leachable study may be justified. A simulated Leachable study is a surrogate study, where instead of using the drug product that's complex, you would use a simulating study, a simulating solvent that mimics the extracting properties of the drug product. So for instance, for blood products, it's not uncommon that sponsors use 40% ethanol in water as a solvent to mimic the extracting properties of blood products. Importantly, simulated Leachables studies require strong justification, demonstration of due diligence, and I would recommend regulatory consultation before committing to this type of studies. Yeah, I think I did this. Oh, sorry, I'm going to the wrong direction. Apologies. So another aspect of the ICH Q3E is that, and this is really important, is that it is a risk-based approach on how to deal with extractables and leachables. And the scrutiny level of testing and evaluation for these impurities is risk-based. So in a low-risk scenarios that could be, for instance, a solid drug, oral drug, that is stored in a container closure that is in compliance with food contacting regulation. So in those cases, for instance, an abbreviated data package may be justified. You can use in this abbreviated package, you could leverage prior knowledge, you can perform quantitative studies only, and by all this needs to be scientifically justified. For higher risk products, such as products that are for using in routes that are parenteral, intrathecal, inhalation products, also liquid dosage forms, and biologics. Those typically requires extensive testing, which includes comprehensive extractables studies, leachables studies conducted over its shelf life, and detailed toxicological risk assessment. So in order to understand the risk regarding leachables, it is important to understand the concept of analytical evaluation threshold, AET. The AET is the minimal amount of extractable or leachable that should be identified for chemical structure, quantified, and reported for safety assessment. In other words, extractables and leachables that are below the AET, you don't have to do anything because it's considered low concern. So how do you calculate AET? The AET is based on the safety concern threshold, SCT, and the maximum daily dose of the drug. The AET is product specific because it depends on exposure. It depends on the route of administration and also the duration of exposure. Very important to remember here is that the AET is the foundation for the extractables and leachables risk assessment and control. And it requires a close collaboration between analytical chemists and toxicologists to set this threshold for your specific product. So, the safety assessment as per Q3E is built on the safety concern threshold, SCT. So SCT is a threshold below which a leachable presents negligible mutagenic and non-mutagenic toxicity. And the SCT is very important because it's used on the calculation of the AET, and therefore the SCT determines if a leachable needs to be identified, quantified, and reported. And so what is the SCT? SCT can be either the threshold of toxicological concern, TTC, or the qualification threshold, the QT, whichever is lower. So the qualification, the threshold of toxicological concern, TTC, are basically covers the mutagens and is derived from the ICH M7. Now the qualification threshold, the QT, this is something that is special on the ICH Q3. And this threshold is applied to non-mutagenic compounds with systemic toxicity. So how did we come up with this QT? So the Q3E expert working group prepared a database containing 330 non-leachables. Based on this database, the EWG used the permitted daily exposure methodology to calculate the PGEs for the most toxic leachables, such as benzopyrene and the bisphenol A. So the QT that are based on this very low, the lowest PGE values, is intended to be conservative and protective of leachables that are highly toxic. So if you go to the draft guidance of the Q3, the table 1 has provides the toxicity thresholds, either the QT, I don't know, both QT and TTC. So table 1 provides systemic toxicity thresholds and the local toxicity thresholds. For systemic toxicity thresholds, the table offers QT for oral, parenteral, dermal, transdermal, and inhalation routes. For local toxicity threshold, the QT is offered for topical, ophthalmic, subcutaneous, intradermal, dermal, transdermal, and inhalation. Of note, there is no QT for sensitive routes such as intracerebral, intrathecal, epidural, and intraocular. And this is not an oversight. Okay. And this is not an oversight. This is because there was no robust toxicological data that could be used for a determination of the QT. The ICH Q3 also provide a leachable classification system, and that is a good thing because it is streamlined the work. So class 1 leachables are the ones that should be avoided. That include many compounds from the AM7, such as AM7 cohort of concern, class 1 compounds, non-mutagens with PDE below the QT. So the action here, if you find class 1 leachables in your material, the recommendation is to avoid using it. But if that is impossible, then compound-specific safety limits should be used. Now, class 2 leachables, those are the ones that should be limited. And it includes ICA compounds that are ICM M7 class 1, that we see acceptable intake above 1.5 micrograms per day. Also include chemicals that are class 2 and the class 3. From M7, and the non-mutagens with PD above the QT. So the action here, you should control the leachables within the TTC or the QT. And then class 3 are the leachables of low toxic potential, and it's defined as having a chronic parenteral PDE of above or equal to 1 milligram per day. So here I'll offer in a minimalist manner some practical implementation ways for when it's time to implement the Q3. First, consider extractables and leachables in material selection, formulation development, and equipment locking. Do this before any manufacturing decision. Tailor your studies based on the specific risk of your product. And always have a scientific evidence for all decisions that you make. It is very important that analytical chemists and the toxicologists work together to get right the determination of the analytical evaluation threshold. Regarding documentation, my recommendation would be that you maintain comprehensive records about anything and everything, and the scientific justifications for all approaches at every steps. Adopt a life cycle perspective, and that means that you establish very robust change control procedures, including vendor quality agreements that will tell you every time that a chemical composition changes or a manufacturing change for that specific material. And since those aspects can change the profile of the leachables, and therefore it may impose a safety concern for your product. And also, I recommend that you engage early with regulators, especially if your drug involves novel approaches, or if you decide to do a simulated study, or if your drug is for using sensitive routes and the very complex modalities. So it's always a good idea to engage regulatory agencies early in the process, so to align expectations. So in summary, the ICHQ3 provides a holistic framework that ultimately integrates quality and safety considerations. It is a risk-based approach that gives you flexibility, and as long as it's scientifically justified. It provides clear safety thresholds, and this is very important. So, it offers TTC and QTs for different routes of administration. It provides a leachable classification, therefore, it connects chemical entity with regulatory expectations. The ICHQ3 establish life cycle expectations. So, and that goes from the development phase through the commercialization and beyond. And it is a global harmonization that provides consistent expectation across regions for both regulatory authorities and the also industry. The bottom line is that the ICHQ3E is focused on patient safety through this comprehensive risk-based assessment, flexible risk-based assessment, and the control of leachables impurities. I do want to take a moment to thank the leadership, or BRR leadership, for their strong support of this work. I want to thank Dr. Ann Eder, Dr. Ujwani, Dr. Wendy Paoli. From CBER, I want to thank Dr. Edwin Zhao and Dr. Dane Mello for their extraordinary efforts on the development of this guidance and also outstanding expertise. I also want to thank the whole ICH EWG for their continuous strong commitment to the guidance, for their expertise and continuous collaboration. And finally, I would like to thank the conference organizers for this opportunity to speak today. Also, I want to thank you all for listening to me. I hope that this overview has been somehow helpful. And I open the floor for any question that you may have or comments.
Thank you. Still in a couple of questions. Question to the audience.
Excellent talk. I'm in the gene therapy area, and my question is about the requirement for leachable studies in that field. So a lot of sponsors are not comfortable with the expenses that they have to go through because these are ultra rare diseases. And of course, you talked about risk assessment, which is a very important part of it. And the question is, if the extractable, which vendors do with different components of direct contact, there are no risk associated with those extractable. And in view of the fact that the prior knowledge exists within the CDMO, et cetera, of leachables and impact on the product quality, et cetera, is it possible to essentially have a scenario where there's, based on the risk assessment, based on the other criteria, that you would not do a leachable study? So it's a tough question, but it's case by case. I understand risk and science base, but can you comment on that?
That's exactly what I wanted to see. It is case by case, but I wanted to clarify a few things. So the ICHQ3E is applicable for cell and gene therapy.
Right.
And in any cases, for any product, you can always use data that comes from the vendors. You can leverage prior use, and the risk assessment is risk-based, and that's why it is going to be case by case. I think the best is for a specific product. I think the best is always reach the agency. FDA has different channels for communication, and you can get answers to your questions through Type B meetings or Q submissions and the other formal correspondences, and you would have that answer better answered than I could provide to you today.
Okay. Can you tell us a little bit about who does the review of the extractable, leachable within different offices or centers at FDA? Generally speaking?
I would like to restrict my answer to what I know. I work for the Office of Blood, OBRR, and in the OBRR, there are pharmacologists and toxicologists and CMC hybrid folks that are the ones that review extractables and leachables. And sometimes it's also the case that we utilize other offices such as CBER-Regulated for analytical inputs on the methods. You're welcome.
Yes, I was just wondering that. I'm just wondering that. I mean, it seems like it's a new guidance, and it seems like it's a CBER product. So I'm not very sure about it. So I believe there is. I mean, even if it's the CBER-regulated products, it should be also be applicable to CDAR-regulated products. Is that right?
That's correct. So the ICH Q3E is applicable to new drug products. It's applicable to combination products. For instance, if it's a device-drug combination, the Q3E is applicable to those combination devices in case the drug is the primary mode of action. Right? But I'm going to give you my scientific opinion about this, and this is not FDA binding recommendation. I think for even standing alone devices could benefit also from the ICH Q3E, but although it's not going to replace the ISO 10993. And so does that answer your question, or?
Yes, I think, I mean,
yeah.
Sort of,
So I mean, the drugs, I mean, CBER-regulated biologics, right? Most, and CDER also have some biologicals, and they deal with small drugs, and it is applicable all across the board.
Yeah. Also, I have another question. Let's see if I have. Oh, okay. So I'm just wondering. I mean, it's for rare disease, and then probably the dose is probably not that high. In that case, also the batch size is really not that much size. So do you think that is under those circumstances, that is, you have this kind of extensible, leachable and extractable studies to be carried out?
So the level, the scope and the depth of your extractables and leachables is risk dependent, right? And the FDA does have considerations for products that are for rare diseases. But I'm not in a position to give you more information about that. I think the best is to utilize one of these communication channels and obtain an appropriate response for that.
Yeah, we have time for one online question. So what are the agency's expectations for leachables that are not included in the current LC or PubChem databases where no prior relevant toxicology knowledge exists and the thresholds such as AET, SCT, or TTT have been established?
So if I understood it, correctly, the question is how to deal with the extractables and leachables for products that don't have a threshold, established threshold. Is this the questions?
What do you do with leachables that aren't described in the databases and where no prior toxicology knowledge exists?
So I would say that, so first of all, according to the Q3E, the leachables that should be identified, quantified, and assessed for safety are the ones that are above the analytical evaluation threshold. So that's the phase 1. That's the step 1. If leachables are above the analytical evaluation thresholds, but then there is no toxicological data or whatsoever, you could try to use the REdI across approach, which means surrogates. So the Q3 does have some methodology, some described methodology on how you could use the surrogate. There are some uncertain factors that are applied to the permitted daily exposure determination, and therefore you can go from there. But in the worst case scenario, when you don't have toxicological data and you don't have the identification of the leachables, I would say that the appropriate action would be to consider the leachables, this specific leachable, as considering the worst case toxicological concern. So it would be, for instance, you consider that this compound is mutagenic, and therefore you could use the threshold of toxicological concerns that are based on the ICH M7. So that would be my recommendation. You are welcome. Thank you.
So now we're going to have the closing remarks for this day. These are going to be presented by Dr. Price.
Thank you. First of all, I'd like to thank the people who stayed in the room throughout the duration of the studies, and particularly I'd like to thank our online participants who bore with us despite some technical difficulties during some of the presentations. I'd also like to acknowledge all of our speakers today, and in particular, I'd like to acknowledge our front room staff, Laurie Warren Henderson and Stacey Rivette, and our backroom moderators who helped organize the online questions, Jingyang Wang and Shen Lin, and we're looking forward to seeing you all tomorrow. I believe there's one more slide. I'd like to remind everyone that recordings will be posted to the event webpage once they are available. That will probably be in a couple of days' time. And a certificate of attendance to support continuing education credits from these organizations will also be available. I'd like to encourage everyone to carry out the evaluations for today's talks and also for tomorrow's talks if you're joining us then. And these evaluations will be available for 2 weeks. And that concludes today's presentations for the CBER-Trek. Thank you very much for attending.